Yeah, so a very good morning to everyone. Many of you might not know my face. My name is Martina. I'm the chair of the FYNBOS Forum, and we are your hosts this year for the Mediterranean Ecosystems Conference. And it's our great privilege to welcome you here this morning with us. I understand many people are still having breakfast, so we'll have a few more delegates in the room later. Yeah, we'd like to welcome our online and our in-person delegates. We've organized this as a hybrid event, and it's also been postponed about 3 times. So we're really happy that you're able to join us and that you've arrived safely. I'm not going to say too much about Medecos because I think Professor Karen Essler will be covering most of it in her presentation or keynote later this morning. So I'm going to cover some of the house rules that will hopefully help you to navigate and have a wonderful experience with us this week in Langebaan and for Medecos 2022. We'd like to encourage everyone to please download the app if you haven't done so already. Many of our communications and messages will be pushed through the app. So please check the links there. You can either scan the QR code or maybe look back on your emails. I think you should have received a link to download on your mobile device or laptop, whichever one you prefer. Rupert has asked me to remind everyone to, those of you that are tweeting, to use the hashtag, is it FYNBOS Medecos 2022? So please do that and help us to gather some people in the online or social media circles as well. Yeah, and there's a website at the bottom of the slide there. Our program, we believe, is quite well outlined on the website. Field trips, symposia, keynote speakers. Everyone should be able to find most of the information there. And if you can't find something, you're welcome to ask us. We've got a 30-pool strong member of custodians that's also able to answer some of your questions. And Madeleine and her team is also on standby. A short thank you. I'd like to thank our scientific program committee for putting together an exciting program that we hope will be engaging for you in the next couple of days. I'd really like to single out Karen Essler again as a really star mentor, if you want, Karen. So thank you so much for your patience and for populating and always being on standby. Especially, it's of this scale, it's a bit more than a local FYNBOS Forum conference. So thank you so much for that. Jasper, also to you, thank you so much. You've been very patient in your new position also. So thanks a lot for helping on that front as well. At this stage, I think it's probably fitting to invite my vice chair of the FYNBOS Forum, Adrian Grobler, to come and stand next to me. Adrian is also helping, obviously, a lot with the arrangements and the flow of the program this week. And I believe he'll be chairing many of the sessions tomorrow also. So yeah, Adrian is an excellent wingman. Yeah, one has to acknowledge, I think the substance on the program, and it wouldn't be the same if we didn't have our 13 keynote speakers. So from the FYNBOS Forum, thank you very much for accepting the invitation to deliver a keynote. And we're really looking forward to listen to all of you during the course of this conference. I don't have the names there because the list is very long, but they're at least organized by day. And we look forward to listening to Karen shortly, followed by Anton Poe and Tony Verboom also a little bit later today. It's also probably suitable to mention that some of our keynotes are online. I think it's Kathleen Kay, Alexandra Seafard, and also Rob Salguero Gomez. So, I mean, there's a bit of flexibility in the format as to how we'll engage with our keynotes during the course of this program. A reminder to please sign up for your Wednesday field trips. We've got 6 field trips listed on the website, so please have a look there and register so that we know exactly how many people to expect for each field trip. Then a thank you to Kirsten. Kirsten has been coordinating the field trips. Kirsten, can you put up your hand? There she is. So thank you so much for that. Kirsten is not able to lead 6 field trips, but she'll be able to put you in contact with the right people So she knows, she has a pretty good understanding of what's going on there. Yeah, and the sign-up links are on the website, so please have a look there and register. Yeah, it's a 5-day conference. It's a full program, so we've worked in some time for rest and relaxation also. There's a Tuesday afternoon yoga session, which is given by Barbara Ziele. It's an online yoga session, and we'll post a Zoom link or make sure that everyone has access to that link. And also Thursday morning, 7:15 to 7:45. So 2 short sessions to relax and engage if you wanted to. Yeah, a huge thank you to our sponsors. There's probably too many to single out now for the sake of time, but we're fortunate to have a really nice collection of sponsors and people who were able to give either, there's exhibitors at the back there in the tea breaks, and we've also got, or fortunate to have had quite a few prize sponsors and yeah, thank you gifts as well. So thank you very much to all our sponsors. A huge thank you to Madeleine and Wetlink. Wetlink is our conference organizer, and Madeleine is sort of at the wheel there. So if there's anything that we can help you with to navigate, or if you have a question, please feel free to approach us. Tina, Robin, Stephanie, sorry, I don't have your name on here, but yeah, thank you, Madeleine. We'll thank you properly a little bit later, but you've made it come together really nicely, and we're very, very happy for all the help and efforts you've put in. So thank you very much. A thank you to our session chairs. So there'll be a couple of people running around to also make sure that the hybrid format runs as smoothly as possible. We've got people on the floor, obviously, and then also checking that the online delegates are also happy. We'll be able to pose questions from both ends. Yeah, and our session chairs, thank you very much. It is quite a job, so thanks for volunteering to assist in that sense. A massive thank you to Adrian Drotsky and his team from Audio Pimps, who's set up sound and lights for us. Thank you so much, Adrian. And also to Charles and his team, the Stream team. Sorry, I got the name wrong, Charles. Stream team. But thank you so much for you guys as well. Yeah, and then the only last little bit of information I'd like to share is that Medecos 2026, I believe, will be in Australia. They've been happy to accept the next round. And other than that, I can't give you much more detail. But it's looking like it might be Perth, so an opportunity to hopefully travel to that side of the world. I think, was there anything else? I should probably say that one important thing is that we'd like you to just make sure that you've booked your dinners. Tonight we'll be at Cape Town Fish Market. Tomorrow evening is at Boesmanland, which is slightly off the venue. Wednesday, I believe, is at Brouwhouse, and Thursday is at Boesmanland again. But please do ask us if you struggle to find those venues. Lunches will be, if you walk out the back, there's a Cape Town Fish Market, and we've designated that as our lunch venue. Yeah, and I think we'll navigate and try to get this mothership off for now. So please join us. I'm going to hand over to Jasper to introduce Karen, our first keynote speaker. So thank you very much.
Thanks, Martina and Adrian. Yeah, for chairing FYNBOS Forum and for chairing and setting up Medecos. I think they need special acknowledgement given that they're both postdocs. I see a lot of vitriol on Twitter about abusing your postdocs, and I think we've got it down to a fine art here. So thank you very much for all the efforts you've put in. Sorry, I thought I'd pushed a button. Oh, I did push a button. Our next speaker is Karen Essler. She doesn't really need introduction. She's very well known as part of the FYNBOS Forum and has supported that for many years, but is also very well known in the Medecos conferences, having been a stalwart for many years, and actually having been the chair of the previous Medecos that was held in South Africa. So that was just a couple of years ago, of course. So I think Karen's going to give us a bit of a talk about Mediterranean-type ecosystems in general, and perhaps a bit of the history as well, I believe. So over to you. Oh, just one last thing. Sorry, there's two different microphones. So for speakers in general, you can either use the podium microphone or the roaming microphone. Up to you. It's a bit hard to see your own slides if you're standing at the podium. So just make it clear to the chair when you come up. And then if you see me dancing and waving around, it's because I want you to stop doing this. So, yeah. Actually, I'll give you that one. That's okay. And then lastly, when questions are asked, because we have online people, can you hold that? Because we have online people, just wait until the microphone comes to you so that everyone can hear you asking your question.
Great. Everybody can hear me. Well, a very great welcome to everybody. It's lovely to see physical faces in the room after so long. We've been planning this meeting for way too long. So I thought that, I'm not going to give you any heavy science because I thought that, maybe just kind of ease our way into this meeting. There's going to be a lot of fantastic science spoken about during these next 5 days, and I'm really looking forward to the conversations, the connections that are going to be made during this meeting. And there's just something to be said for actually face-to-face conversations rather than sitting in front of a computer and communicating that way. So that was my title, but I guess one could just as easily have kind of substituted the title for that, Partnerships and Collaborations. And the whole theme of this meeting is Partnerships for Global Change. So I thought I would kind of weave in the little bit about the theme around a little bit of the history of some of the kind of partnerships that have been going for significantly long periods, and reflecting a little bit about those partnerships. So the idea of partnerships is something that's really taking a lot of traction lately. This is the Intergovernmental Panel for Biodiversity and Ecosystem Services. I probably got that slightly wrong, but I never get it right. The acronyms are my worst. But essentially, I was involved in the global assessment, which was all about trying to kind of find pathways to sustainability at a global level. And this is one of the papers that came out of that process. It recognized that if we are to effectively address the global change challenges that we are facing, and they are significant, I think there's nobody in this room that would not recognize how terrifyingly significant these changes are happening, and the pace at which they're happening at the moment. But essentially, this process recognized that if we are to effectively change things, we really need to focus on transformational change now. It's no good just trying to scale up what we've already been doing. It just simply is not sufficient. We have to find levers, places that we can actually lever and leverage points which will affect greater transformational change. And what's interesting, if you actually have a look at that figure, is how much of that revolves around collaborations and partnerships. It's incredible. So, things like coordination across sectors, preemptive action, adaptive decision-making, unleashing latent values of responsibility, a whole range of these things require us getting together, talking, and creating synergies that ultimately end up in action. So that's a message that's coming out of the global community. And the partnerships are recognized so much so that we actually have our own sustainable, it actually has a sustainable development goal of its own, the Partnership for Goals, SDG 17. Really also recognizing this point, the same point that was made on the previous slide around multi-stakeholder partnerships being absolutely critical to affecting change. And there's a whole range of reasons why that would be the case. So mobilizing resources, trying to figure out partnerships that will elevate and allow things to happen, creative thinking. There's going to be a lot of that, hopefully, in this conference, especially around the tea and especially around the pub. And the disco, don't forget about the disco. There's a lot of creative thought that goes into that too. So that interaction is really critical. And also contributing to wider agendas, strengthening roots and so on. So really, really critical. The SDG folks have actually got this partnership guidebook that's underway, and they really talk about how we really need individuals with partnering knowledge and skills, but we also need to have institutions that are fit to be able to take on those partnerships and run with them into the long term. So I thought that I would actually reflect a little bit on three of these partnerships, and they just happen to be sort of essentially us, the FYNBOS Forum, the Medecos meeting, and then also I thought if I had time, one of a local partnership that I've been involved in, and took a little bit of inspiration out of this paper, which was really thinking about this how to facilitate partnerships and what are some of the enabling and limiters, essentially, to trying to achieve partnering. And without going into a lot of detail on that slide, these are the things that came up in that particular that paper. The ones in green are all items that can be derived out of Elmquist's translational ecology, so essentially making the connection between the knowledge and the doing. Those were the key things in the light green: collaboration, commitment, process, buy-in, communication, engagement, decision-making, context. But they also added two others in that paper, and this is what I thought was quite interesting: innovation and robust science. I think that's really quite I think a central theme to a lot of the work that we do in the FYNBOS and in the general Mediterranean type ecosystems in general, is that we've got this incredible system that we actually are focusing on. And interestingly enough, a lot of innovation and thought has been derived out of these communities. And I think that's the kind of glue that keeps everything together. As does, maybe I'd have one last one, and that is people prepared to put up their hands and do something to actually make it happen. And so I want to say thank you very much to Martina and Adrian and the custodians, because essentially we made this happen. Finally, it took a while, but we made it happen. And that's because people prepared to put their hands up and make it happen. So, yeah, I thought I would just reflect a little bit on the three different partnerships. I'll talk about our global partnership, or collaboration, if you will, Medecos. It's been going since 1971. Regional, just about as long, 1979 it started. And then I want to reflect a little bit on a science partnership that I'm involved with, and some of the folks in the room are also involved with here, which is really around trying to make sure that the science that we do is embedded into action. So very briefly talk about those things. So let's start a little bit with Medecos. And I thought I'd put this list up. These are the Medecos conferences that have happened over time. The first conference was initiated through a conversation of people in a room. Essentially, it was De Castri and Hal Mooney, Francisco De Castri and Hal Mooney, and that's a photograph of them at the bottom there. Hal Mooney, believe it or not, I think he's still going strong. That man's incredible. But they were interested, they picked up on the interesting parallels between Mediterranean-type ecosystems around the world, and they were interested in trying to understand some of the drivers of these, and ask the question, is it climate, is it soils, what's driving these systems, what's generating the innovation in these systems. So they raised some funding, and the first the first inaugural meeting is sort of largely thought to be held in Chile, in Valdivia. And you can see the themes of the earlier conferences were really around trying to understand these drivers: fire, convergence, nutrients, and so on. in around about sort of 1980s, there was a semi-formalization of Medecos through ISOMED. That's what the name was, the International Society for Mediterranean Ecologists. But that was pretty much about as formal as it actually remained. Essentially, it was a group of people that included Hal Mooney, also Phil Rundell came in very early into the core, and he, in fact, has probably been benignly sort of making sure that these Medecos meetings happen ever since. In fact, we've been in communication together to try and solicit the next venue or next hosts in Australia. So that was a result of a bunch of emails that went around just before this meeting. So that's a kind of informality of this process, but it's resulted in a range of very interesting outcomes. The conferences in bold are the ones that I attended. So I was a postdoc in California in the 90s, just before our independence here. I went to California, voted, in fact, actually in California for South Africa. Then the rest, I started going to these meetings, and they actually were my point of call. And I sort of reflected on why is it that was the meeting. It wasn't ESA, it wasn't the bigger other international meetings. This was the one that I wanted to go to. And I think the reason is because of the people. It's the connections that I made in those early days, and I've continued interacting with all of those people. And so that really, really set the scene for me to be involved in further. And as Jasper said, for my sins, I was involved in the Medecos 2000. I think it was about 5 years into being an academic at that time. I'd just been appointed at Stellenbosch University. And here we are. Should have been 20 years. These conferences go, the idea is that they go from region to region and they sort of return to the same region every 20 or so years. That's the idea. So hopefully in another 20 years, we'll still have this momentum going, and it'll come back to the Western Cape or to the Eastern Cape, wherever it goes next. So that's the... Well, the other thing I wanted to point out about the slide is how you see, as the themes change of the conferences, you can sort of see it reflecting what's happening in society as well. So this idea that things are becoming a lot more complex, human-driven scenarios for evolutionary and ecological change in Sevilla in 2017. And now we're talking about partnerships. It's the social elements are coming in. They're coming in a lot more, as we need to be able to understand this complex social ecological system that we find ourselves in. So the Medecos has been going for 51 years. It's remarkable. And there have been a whole range of outcomes that have emerged out of these meetings. These are just examples, there are more, but meetings, conferences, collaborations, edited volumes, research activities. So, ideas emerging at meetings and people running with them. And above all, a network of shared research, education and practice experience, including some very practical things, like the one was the sort of like focus on biological invasions, which really kind of, I think, sort of launched our sort of research program in that area, both locally and abroad. And around about that first meeting, Fred Kruger actually was appointed onto the ISOMED committee at that stage. So he was the South African representative. And there was a meeting in 1980, this meeting in Stellenbosch, one that I wasn't around, just before my time. I was still a bright-eyed and bushy-tailed student at UCT at the time. But I love this quote that Fred has, because it kind of sums up a lot about what these meetings are about. He said, there was an excitement about the scientific ideas being explored. Mediterranean ecosystems were seen as a natural laboratory for the idea of convergence between different regions. Intrigue arose from the comparative work being done between California and Chile. There was curiosity. It was all tied up with a common culture that included good wines. You have the impersonal institutions, which will continue to exist, and then you have the personal. The fruit lies in the fact that people know each other. The fruit lies in that, and I think that's so true. My professor would write me introductions to people who would never have been seen, I would never have seen me or never seen me otherwise. And I hope to think the same thing has happened with my students and for the other, that's the case. That becomes a way, a sort of a vehicle into this world of research. And certainly, I would say that is my own experience. So very briefly, I got involved since 1994 and then got involved in the ISOMED committee. I think that this was sort of benign dictatorship at the time because I knew that I would run with the conference in Stellenbosch in 2000, which I did. And now here we are back 22 years later, and I'm still alive. It's amazing. But I also met a bunch of fantastic people along the way, and I interacted with those people and have published with those people and have produced a range of research papers with these people. And ultimately, I think kind of for me, a great achievement or outcome was this book, The Biology of Mediterranean-type ecosystems, which was a collaboration between Anna and Brandon, who you can see on that photograph, who I also met through the Medecos community. So it's like a sort of a kind of a passing on. Cheryl Swift, you see there, Phil Rundell, Byron Lamont came for a sabbatical. So these are people that I have met along the way through the Medecos meeting and that have been my colleagues ever since. So one of the products from Medecos was we decided that we'd ask people, if identify questions that would have a high probability of achieving success for conservation in Mediterranean-type ecosystems. And this is the global paper. Jasper was involved in it, as was Nikki Alsop. And that we were the regional representatives for this global work. And what's quite interesting about this, the research topics that were perceived of the greatest importance across all these 5 Mediterranean-type ecosystems and also across stakeholder types. Right at the top, governance, and then if you can see there, public participation, very high up. Obviously concerns about climate change and species management too, but very high up, governance and public participation. It's all about trying to facilitate these collaborations, these networks. And so this paper really highlighted that importance of interdisciplinarity, so large variety of people connecting, and then also understanding the human and social dimensions of any system. So if I was to reflect on Medecos and what has made it continue on for so long, I would say that probably it is this kind of commitment, this idea of this really fascinating system that we're working on. And that actually is probably at the core that's fueling all of this. But it's also about people really, really keen on making it happen and making that communication happen. So hopefully it will continue to last into the future in this complex world that we're finding ourselves in. So there is, I don't know, Sierra in the room. There we go, Sierra. Maybe you could just stand up, but Sierra is actually here. She is wanting to meet, if anybody has been involved in any of the Medecos conferences before, she's interested in recording interviews that capture your memories and perspectives, because that's my little history of Medecos, but there are many other histories. So please actually speak to Sierra. So very briefly going on to the regional partnerships. FYNBOS Forum been going for 43 years, also a learning network. And at the time, in about 1980, Hal Mooney commented that he didn't know of another organization that did quite the same as the FYNBOS Forum did, in that it actually was linking managers, landowners, researchers, planners, in this space, in this creative space to ensure the conservation of the FYNBOS region. And there's a lovely book that was put out by Julia Wood and Caroline Gelderboom, sorry, about that. But that actually records some of that history. And Sierra, maybe that's something that you would be interested in taking a look. But we've managed to have annual meetings for 43 years, going from east to west a lot of the time, and sometimes in small little towns. again, a huge amount of achievement has come out of that. We've sustained this knowledge network for 43 years. We've built a knowledge base. We have built capacity through the students that have attended these meetings, collaborations, edited volumes. So there's a range of fantastic outcomes, including some initiatives like Working for Water, for example, the core of the idea coming from this forum. And if I look at my own experience, I kind of wasn't involved in the FYNBOS biome project in the early days. I was in the Karoo, but I moved into the FYNBOS when I had my daughter and I wanted to be closer to her. So I didn't have to move so far. And again, the same thing. It's those connections that are made. And in fact, also international connections, like the connections with Frank Scher and co, who are also here, and the wonderful work that has been involved in. Two of those papers, the Challenges of Invasive Alien Plant Control and the FYNBOS Proteaceae as Model Organisms, were outcomes out of FYNBOS Forum workshops. So exciting. And my own little sort of contribution was also a FYNBOS Forum initiative, the FYNBOS management book, all around trying to kind of make sure that the custodians of the FYNBOS are taking care. So lots of exciting work, and that's emerged out of these connections that were made. So we also did a local, out of that initiative, we did a local question. We asked the same thing, asking, if you identify questions that would have a high probability of conservation success. And what's, again, probably less illustrative here, and I'm not sure you can see, but the little red numbers are the sort of numbers of questions that are focused in different arenas of this. This is the Intergovernmental Panel conceptual diagram. And again, slap bang in the middle, you see that institutions and governance and other indirect drivers. And that's where most of the questions lie. Again, highlighting the value of partnerships, the value of understanding how governance works, institutional arrangements, and so on. So, also landscape level. So landscape level really does require us bringing in partnerships as well. So there's this great history of the FYNBOS Forum, and a lot of sort of key elements of this network, this spirit of cooperation which has continued to today, the idea of capacity building, diverse learning networks, and so on. And if I was to reflect again as to what benefited the FYNBOS Forum, I would say, again, it's the fact that we actually have this incredible system that we're working on, but we also have the people, we have the champions, we have communication, process buy-in, and so on, from the range of people that keep this going. Very finally, the local partnership that I have, I'm very excited about. That's been going for 12 years, and that's phenomenal for me. This is a long-term research partnership that involves researchers, planners, and managers. And again, there have been some really nice practical outcomes. For me, this is a way of embedding what I do in science into action. So seeing some of the work that our students are doing actually taking place in the environment, partnering and learning with the institutions that are actually on the ground. So again, I think these connections are really critical for us to enable action. And we are going to be, Alana Rabella and I are going to be hosting a FYNBOS restoration workshop tomorrow. And so anybody who's interested in coming here to understanding those connections between partners, research and partners, we would like to see you in the room, and we're going to have a fun workshop tomorrow. So again, if I actually reflect here, it's really around sheer, for these partnerships to work, we have to get the money, we have to keep on going. It's commitment. It doesn't come on its own. You have to keep on pushing to make things happen. So I'm going to end up here by saying that, we have these uniquely special environments, rich biodiversity, deep human connections in these landscapes. We are facing huge global change challenges. We have set ourselves up in the past to be a model system for comparison. I think we can still continue to do that. And one of them is to understand how we have managed to maintain these unique long-term partnerships for really significant lengths of time. I don't know if we can find similar examples at a global scale of any other system doing the same thing. I'd be interested to know if there are. So through these partnerships, we've actually generated cutting-edge ecological and evolutionary research, and we've put a spotlight on the threats, and we've actually have figured out ways for mutual learning and on-the-ground outcomes. And so if we actually are going to continue into this future, which I think is a little bit different to the past in that now there's many things coming at us at the same time, all at once. So it's not only the power situation, it's the financial situation, it's the fact that we have storm surges. All these things are happening. The Anthropocene is on our doorstep. If we are to continue, we need to think a little bit more flexibly, I think, about this. And so those long-term, one of the features of these long-term partnerships is flexibility. And we made this Medecos meeting happen finally, and FYNBOS Forum live, it's happening. So yeah, I think that we need to start reflecting on that. And one of those ways is through understanding our past and understanding what worked, what were the enablers, and what were the limiters, and trying to go forward on that basis. So I think I'm going to end there and say thanks very much. And yeah, over to you if there's any questions. I'm not sure if there would be in this particular historical.
So just a reminder, if you have a question, just wait until we get to you with a microphone, because the online people won't hear you if you're shouting. Any questions?
Stunned silence. Maybe I could actually ask a question as well. Can you put any hands up of how many of you have been to another Medecos meeting? Is there any? Yeah, so there's a fair smattering of Medecos folks around. Yeah, fantastic. Okay, and then the FYNBOS Forum folks, anybody been to a previous FYNBOS Forum? There we go. So, yeah, you all know each other well. There's a question over there.
Hello.
Hi.
Thank you for your presentation. I have a question regarding if you have any tip or any advice for, some of us are not only researchers but also teachers at universities. So I would like your opinion on how to engage young people who are studying biology or environmental science in these type of issues and collaborations and partnerships from early age.
So the way I did it was just made it happen. So luckily I actually have a permanent full-time job, and that's actually one of the things that's changing now, is that there's the permanence, there's a lot more sort of temporary employment, contract employment, and that worries me a bit because the permanence of my position enabled me to build up funds and enabled me to support students all the time. So for most of these meetings, if I don't know, we can probably not now because we couldn't afford to get the car on time here. That's a resource problem, but some of my students are making their way up this morning. But it's around actually trying to raise the funds and getting the students to these meetings, because once they're at the meetings and they're introduced to other people, that's where the connection's made. And in fact, actually in the custodians meeting, I think it was Mashudu who said, please, when we are at the meeting, we need to connect with those people that might be sort of at the edges, because we've got to bring them in. I mean, over the course of the FYNBOS Forum, we've seen a big change. In that book, there's a social network analysis of changes have happened over time. And we've seen how, it's increased in its diversity, it's increased in sort of a range of different, people that have attended, some interesting differences or variations. And most of that's, I think, it's just an ethic, it's an ethos. It's around, I think that's what it is. It's commitment, an ethic, and an ethos, and somehow you have it. And we've got a bunch of fantastic people in this room who do have that ethic and ethos, and hopefully that will mean that we'll continue on into the future meeting like this. So I'm not sure if I know it's challenging, especially in this resource-scarce environment. It was really hard for some people to get here, to fly all the way here. Hopefully the remote or the virtual thing will open up the options a little bit too. So maybe this hybrid version is something that we can think about in the future. It's not easy for the organizers particularly to do, but yeah.
Time for a quick question if anyone has? No? Tony. Mic coming behind you.
Hi, Karen. Thanks very much. So I'm just wondering, you mentioned there at the end the sort of remote, the virtual sort of component of a meeting like this. It's challenging for the organizers, and I think, hats off to them for doing that. I also think that clearly this meeting, the arranging had to start happening during quite an uncertain period. But I wonder about looking forward. Is it the plan to carry on having virtual components? Because to some extent, I wonder whether it doesn't create a sort of a way out for people to. I understand, obviously, it improves access for all, but at the same time, a big part of a meeting like this is actually having people hear the face-to-face experience, and it's quite easy, or it provides an incentive for people to stay at home and rather just attach virtually. And I wonder whether something isn't lost in all of that.
I tend to agree that it's much better for us to be face to face. I mean, look, the reason for this one is really because of the uncertainty. So to be flexible, an institution that's going to be long term has to be flexible, and we needed to figure out a way that we would still go, knowing that. I mean, we have had cancellations in the last little while because people couldn't travel because of travel embargoes. So we really wanted to actually make a way that they would I think in the future it's going to depend on who organizes those meetings, because the liability is taken on by the local organization that runs this. There's not a pot of money that follows Medecos around. It's the people that follow Medecos around, not the. So I guess it depends on the organizers and how they manage that. Yeah, there's a question from Rupert.
Can I respond for you? Yeah, I mean, I think Rupert was just making the point that the access for all thing is huge, and I found being able to participate in big international meetings remotely has been amazing for us, who sit like many hours of flight away from these conferences and things. But it is a trade-off. You definitely don't get the same out of it if you're online versus in person, and it makes it harder for the people who are there in person. So hopefully, as we practice this, we can get better, and we could always maintain a bit of an online presence. It would be, I feel, would be great, because the access for all thing is huge. I think it's the difference. It's other youth and the disabled people, everyone else. Yeah, youth, disabled, if you can't afford it, that kind of thing. It literally was like 10 times more expensive to get there and actually be there in person. But you probably, at the moment, are only getting 10% of the experience. Correct.
And I also think there's a generational thing here. I mean, like, maybe us, you're in my generation, Tony, but, like that sort of whole thing, we went a massive learning curve under COVID, but I think the others, they were born with a cell phone in their hands, They know how to do this. They don't see the difference. So I think there's a generational thing, and that luckily we have that generation taking over. So the techo generation, they can handle that.
Well, thank you very much, Karen. That was a great intro. So we're going to move on. I should just say, for the conference app, if you can ask questions through the app, even if you're here, and for the online participants, if you click on the session that you're in, there's a button that says "Ask a Question." So you can ask questions through the app, even if you're local, and I'll try and keep up with what's going on in there and what's going on in the room. But we're going to move on to our next speaker, who is Professor Anton Poe from Stellenbosch University. Sorry. Thank you, Karen. Can we all... So our next speaker is Professor Anton Poe from Stellenbosch University. And he's going to tell us a bit about the ecology of the area that we're in, specifically in the west coast of the Cape Verstic region. Anton has a long history with this landscape, both personal and in terms of research. So when he put forward just a short talk about this, and then we instantly thought, hey, hang on, well, what are we thinking here? We need to have Anton give us a bigger introduction, and especially for those who aren't familiar with the region. So, Anton, if you're...
Right, thanks Jasper, thank you Karen and all the organizers. It's really a fantastic contribution that you make. So the aim of the talk that I'm going to give is really to introduce those of you who have just gotten off the plane to the West Coast region where we are having the conference, and maybe also for those of you who are online to wish that you were here. Hopefully you'll get to be here soon. So the West Coast is culturally a very clearly defined concept. Small-scale fisheries in the lagoon, some of the best cold water surfing in the world, but don't tell anybody. Cute little houses with roofs thatched with Restianaceae, and some of the best rock art in the world, which is the legacy of the Khoisan people. Biologically or ecologically, the region is not very well defined. So when people talk about the West Coast, they usually either lump it with the greater FYNBOS region. Whoa. That's the pointer, I think. Have I got a pointer? Yes. So the West Coast is usually either lumped with the broader FYNBOS region, or it's broken up into its different vegetation types and then treated as separate units. So I thought maybe one way to deal with the area is to talk about the West Coast municipality as the region of interest that I'm going to discuss today. And it seems like an odd choice for a biologist. It has one advantage, which is that you avoid that kind of circularity that comes from defining a region based on ecological characteristics and then turning around and concluding that it has those characteristics. So in a certain sense, the region is defined free from those constraints. If we look at the region topographically, it's dominated by this broad coastal plain, which then rises up to the Cederberg Mountains in the east. The sea, as you'll notice, is quite shallow around here. So the question I want to address in the talk is kind of a broad question, and I must admit this is not going to be a data talk, but the idea is basically to say, well, what kind of species do we get here on the West Coast? And what are the processes that shape those communities? So the West Coast we can think of as one great big community. And maybe one scheme that I find useful is this one where you start with an original community, like this one here. Species can be added to that community through speciation. Here we've added the yellow species. Species can be added to that community via dispersal. Here we've added this dark blue species. And then random sampling, which is an inevitability that happens with every generation. Well, the next generation is a very small sample of the vast number of offspring produced. So which species are going to be left out? Well, in this case, you can see the green species was left out. So the sample, which is the next generation, is simply too small to represent the diversity of individuals that were present in the previous generation. And then lastly, selection, which is a word that maybe you're not familiar with in an ecological context, but it's really the idea that certain species, based on their traits, are going to perform better than other species, and they're going to become more common in the community, they're going to take over the community, and they're going to come to dominate that community. So this is really where most of ecology is located, and so I'm going to be speaking mostly about selection. Which traits do species have in certain environments that allow them to thrive there and allow them to dominate that community? So this correlation between the environment and the traits of species. So what are those agents of selection that are going to produce this effect on the community? Well, it's the usual suspect. So precipitation, nutrients, fire, herbivores. Nothing new there, I guess. So let's start with precipitation. In a broad view of Africa, you can see the region where we're located, the West Coast, is dry. You can compare that with the East Coast. You can see it's a little bit wetter along the East Coast. So this part of the world spends the summers inside the Hadley cell. Hot air rises at the equator. Moisture condenses, dumps all the moisture at the equator region. That air moves southwards at high altitudes and descends as dry air around 30 degrees south, where we are now, contributing to the dryness of the region. And then that air has to return to the equator, so it returns to the equator as a south wind, which gets deflected to the east and becomes a southeast wind because of the rotation of the Earth. So the summer conditions here are dominated by these southeast winds, and you might have noticed that there's a thriving kiteboarding industry here in Langebaan. Those southeast winds bring a little bit of moisture to the east coast of Africa, which make it wetter on the east and the west. And then in the winter time, these Hadley cells move a little bit to the north, and we start to feel the influence of the feral cell, which has these westerly winds that bring a bit of moisture to the west coast and make this a winter rainfall region. You can see how critical it would be if the climate change predictions, which are that the Hadley cell will increase in size, what effect that's going to have. It's going to push the feral cell further south, and we won't get those westerlies anymore. So that's a scary thought. If we look at a map of rainfall seasonality, you can see that we are very squarely in a Mediterranean climate region here. So the blue here indicates parts of South Africa which are receiving more than 4 times the precipitation in winter as in summer. Intriguingly, the FYNBOS biome extends as an L shape off to the east here, well outside of the winter rainfall region and into the all-year rainfall region. And if we look at the amount of precipitation, if we focus on the west coast region that we are discussing, you'll see that there's a very steep gradient in rainfall, dry on the coastal plain, maybe 250 mils or so, going up to about 1,000 mils in the high peaks of the Cederbergs. So the geology of the region is fascinating. These maps always look like a pot of melted crayons to me, but we can try and just do a quick overview. The nice thing about geological stories is that they're always chronological. So let's start with the oldest rocks. The Malmesbury shales, which are shown in this sort of light brown color, they are very common on the coastal lowlands. They erode into clay soils. They are ancient, Precambrian, 550 million years old. They contain practically no fossils, partly because they're metamorphized, but also just that there wasn't much life at that stage of the game. There are granites indicated in this sort of maroon or reddish color, which have intruded into those shales, and they are the same age as the shales, also Precambrian, 550 million years old or so. And then overlying these shales and granites, we see these purple sandstones of the Table Mountain group. They erode into sandy soils, nutrient-poor, acidic soils. And then overlying this whole lot, we get these quaternary sands which have obscured the clay soils on many parts of the lowlands here. So sandy soils. Along the coast, the quaternary formations are limestones, calcretes, all very recent. So if you think of the geology of this region, it's kind of like a big book, and you've just ripped out the whole middle section. You've got like the very first Precambrian page, and then the very last page, these very recent calcretes that are thousands of years old. So we're here to talk about the vegetation, so let's do that. Here's a vegetation map for the region. And what I'm going to do essentially for the rest of this talk is to do a quick little tour. So we're going to start over here in the mountains, in the FYNBOS region, in the Cederberg Mountains, and then we're going to, something looks a bit odd here, there we go, I'm here. In the Cederberg Mountains in the FYNBOS region, we're going to hop over to the Renosterveld on the shales and on the granites, and then we're going to end up where we are now, which is in the Strandveld on the coast, on those calcretes and more alkaline soils. But while I've got this map up, notice that the FYNBOS, which I'm going to discuss first, is not only in the mountains over here, but it also extends down onto the coastal plain where those recent sands have obscured the shales. So this is a little bit of FYNBOS up in the mountains, not far from here. And this is a typical heathland. So if you follow Specht's classification, this is a heathland, and it would group with the heathlands of Scotland and of Borneo. The Proteaceae are the dominant shrubs here. This local endemic from the Cedarburg is a real beauty. Many, many localized species. Many bird-pollinated proteaceae. And then the Restionaceae are a very important component, perhaps one of the most defining features of the fynbos region. Shallow-rooted, they survive the summers by virtue of the fact that they're basically leafless and they've got these very hard culms. The proteas can survive the dry summers by virtue of the fact that they've got very deep roots and can still access some moisture during the summer. And then the Ericaceae, which make it a heathland, are extremely diverse, maybe as many as 500 species. Microphyllous, you can only just see the leaves there. Whoa, happened again. Very small leaves. Those little leaves are good for cooling the plants down in the summer months without even transpiring. They can lose heat, and that is a way of coping with the Mediterranean climates. A nice example of a plant that deals well with the low availability of phosphorus is Roridula. It's in this endemic family, Roridulaceae. It's one of the, 5 or 6 or something families that are endemic to the fynbos biome. And this one is carnivorous. It captures its prey with sticky leaves, but it can't digest them. And it actually depends on these mutualistic bugs to digest the prey for it. And then it absorbs the phosphorus, etc., from the feces of the prey, from the feces of the bugs. So a consequence of having very nutrient-poor soils is that the landscape has very few herbivores, big herbivores. Biomass accumulates. That biomass eventually gets consumed by fires in those dry summers. So here you can see that many of the plants are serotinous. This protea has cones that are opening up after the fire, releasing the seeds into this cleared landscape with a little bit of nutrient available from the ash that was formed by the combustion of the adults. And almost all fynbos plants have their reproduction timed with fires in one way or another. One way to do that is to have fire-stimulated flowering. So many of the geophytes, and this is another common growth form in fynbos, is to flower only in that first year after a fire. And there are a great many plants that do this. Many of the orchids flower only 1 year after the fire, and then for 10 years or 12 years, you don't see them above ground. They make leaves in winter. Summertime, there's nothing above ground. And so this geophytic lifestyle where you can be a bulb underground is a good way to survive in a Mediterranean climate region. So the region is very strongly controlled by fire. The openness of the environment, the biomass, the height of the vegetation is all determined by fire. And it's really the exception that proves the rule. So this is a cedar tree, an endemic Cypressaceae in the mountains close to here. And you can see that it can grow into a decent-sized tree, big biomass there, proper tall tree. And it's doing that by virtue of the fact that it is growing in a fire-free microclimate in the middle of all these rocks. So if you can keep fires out, you could potentially get a lot more biomass in this region, despite the low availability of nutrients. Okay, let's move on off the mountains and into the coastal plain. So we are going to be looking at the Renosterveld, this blue region here. You can see that it's growing on clay soils derived from those granite boulders in the foreground. It's a gentle, undulating landscape. And these are, I think, what would be called Mediterranean shrublands by Sprecht. There are no Proteaceae here, no Ericaceae, no Restionaceae, or very few of all of those. Asteraceae are the dominant shrubs. The soil is more nutrient-rich, and that has many consequences, as we'll see. It's also a fire-prone system. And after fires, we get a lot of annual growth. These are all Scrophulariaceae. Relatively few annuals in fynbos by comparison. There are also many bulb plants in the Renosterveld. So again, a good way to survive the dry summers. And many of these bulbs, in fact, all of them in this picture, will flower only in the first spring after a fire. These happen all to be in one clade, the oil-secreting orchids. And many of them, in fact, I think all of them, are endemic to Renosterveld. So a consequence of having a little bit more nutrient available is that these regions have large herbivores. Historically, there would have been big herds of herbivores on the lowlands. These Bontebok, for example, are endemic to Renosterveld. And then a sort of a sad consequence, I guess, of having some nutrients in the soil is that this is the breadbasket of South Africa. So if we look at a map that shows landscape transformation, you can clearly see where the Renosterveld lies. It's this region here, which is completely transformed. That's what the white indicates. And so Renosterveld today exists only as these very, very small fragments, big series of very small fragments. In comparison, the fynbos, which lies over here, is relatively untransformed. It's maybe a good point here at which to make some reference to random sampling, because in the discussion up to now, I've been pretty much following the logic of, well, the environment looks like this, it selects for certain traits, and so we get a community that looks like that. That's very deterministic and trait-based, but if you walk up to a small fragment somewhere here in the Renosterveld, you'll find that certain species are absent there. And the reason for that would simply be due to the fact that the sample you're taking is very small, and that sample can never represent the diversity of species that are present in that landscape. So that would really be an effect of random sampling. And you'd be wrong to conclude that those species are absent because of the environment. They're absent simply because the sample you're taking is so small. And I guess the moral of that story for me would be that if you want to conserve the diversity in that landscape, you have to conserve all those fragments, because each of them is a different random sample, and together they might conserve the whole. Okay, we're going to end off the vegetation part of this talk by talking about strandveld in red here, along the coast. So this is growing on those calcareous soils that are more alkaline, that are richer in phosphorus, enriched by the sea. Aerosols from the sea are bringing nutrients in. This is the kind of vegetation that dominates the West Coast National Park, just to the south of us. And this is a very different world. This vegetation, again, lacks those typical families of the fynbos region. There are 4 species of restios only, no proteas, no ericaceae. The plants have quite big fleshy leaves quite often. Some of the families that are dominant here are things that you might not expect, like Celestraceae. There are many species in the Celestraceae. These plants are, in a certain sense, almost forest plants. So many of these same families or genera, even species, along the east coast of Africa would form trees. And the ecology of this region is very forest-like in a certain sense. So the plants here are almost all dispersed by birds. They have fleshy fruits. In contrast, fynbos and the Renosterveld have small dry seeds that might be dispersed by ants, have eliosomes in fynbos at least. This is a very different environment in that sense. Many plants are deciduous, which is also something that you don't see that much in fynbos, at least in the shrubs here. There are many deciduous shrubs, and there are very few deciduous shrubs in fynbos. So that's a way to cope with the dry summers. And I guess the larger availability of nutrients means that you could make new leaves when the winter comes, whereas you can't easily do that in fynbos or in Renosterveld. There are also many annuals here, like they are on the Renosterveld, but these are not fire stimulated. They flower in the open spaces between the bushes, and they're stimulated to flower and to grow by rainfall, not by fire. The nutrient availability attracts large herbivores, and they play a big role here. They open up the vegetation, and I think to some extent they contribute to the formation of these bush clumps, which are very characteristic of strandveld. So these are multi-species clumps. Some of the nurse plants are spinescent, but others are not. It's just very nice for a bush to grow in the middle of these clumps because there's an accumulation of leaf litter. It's quite moist in there, so in the mornings the sea mist condenses on the plants, and there's a lot of precipitation which is not measured that you find inside these bush clumps. Protection from herbivores, protection from wind. So there's an interesting landscape in the sense that it's dominated by facilitation rather than by competition. But notice that there are also these open spaces between the bush clumps where other species grow, light demanding species like Brunsvigia, which has big flat leaves right on the ground. It flowers in autumn, just before the winter westerlies hit the Cape. It's pollinated by sunbirds, by malachite sunbirds in particular. Produces these recalcitrant seeds that need to germinate immediately. So the inflorescence breaks off and rolls across the landscape, scattering those seeds into the open spaces between those bush clumps. And then it survives the dry summers as massive bulbs that you can see over there. They can be as big as your head, some of them. So look out for them. So this time of year, they'll look like this. You'll see those big leaves on the ground. They're very common in the West Coast Park. You can also be a bulbous animal. So these rain frogs, I don't know, you might have heard them last night. They were driving me nuts. This one is endemic to this West Coast region, and they survive the dry summers by being underground as well, and never coming up at all until the rain starts in winter. They also have no tadpoles. So a discussion of the ecology of the region would be incomplete without mentioning the marine environments, but I'll only do it very briefly. So you are probably aware that the Langebaan Lagoon is a mecca for the birds in this region. So most of the wading birds and the terns, for example, also that visit this area in summer, breed in the Arctic and then undergo this massive migration to come and feed here during our summer. And the summers on the West Coast are particularly productive because those strong southeast winds blow the surface waters offshore, and those offshore, that water that moves offshore gets replaced by deep water that's nutrient rich, producing a bloom of life during summer when the terrestrial environment is completely dry. So this, I think, is a little video which is going to show you, hopefully, that we've been moving across the landscape from one vegetation type to another. Can someone push play? It doesn't matter if it doesn't move. What I really want to do is show you that we've moved a short distance across the landscape between different vegetation types. But if we take a longer jump to a different environment, for example, one that's also in the Mediterranean climate region, but is located a long way away, like southwestern Australia, we encounter a landscape that looks remarkably similar to the one outside the conference venue here. So this is Margaret River in southwestern Australia. Superficially quite similar, but the species and genera and often even the families are different. That gave me reason to think a little bit about how such differences arise. And in this sort of conceptual diagram here, I've shown similarity between two communities as blue. So dark blue means that they're two communities that are very similar. And you might get two communities with the same composition if you are geographically very close together, or environmentally very similar, or both. But as environmental similarity declines, communities become less similar. So as we moved from, let's say, Renosterveld into FYNBOS, or if we make an even more distant comparison between the West Coast and the rest of Africa, we find they're very dissimilar because the environmental distance is great. The rest of Africa is dominated by C4 grasses, summer rainfall, and we're finding great dissimilarity between the communities in the West Coast and the rest of Africa. If we compare Margaret River or southwestern Australia with the West Coast, we find that they're environmentally very similar, but geographical distance is great. So dispersal limitation is what's causing the differences between these two sites. And the importance of dispersal was really brought home for me by a picture like this one. So I think you're going to know most of these plants if you're from the Cape. This is Senecio elegans, I think, and Tetragonia. That green stuff in the background is Pelargonium capitatum. The snail in the front is Theba, which is very common around the conference venue here, but it's from Greece. But the photograph is taken in southwestern Australia. So you can see how many of the South African plants do wonderfully in the same environment if you overcome dispersal limitation. And I think the take-home message there is that those of you who are going back to other Mediterranean climate regions, throw away your socks full of seeds before you get on the plane. if you walk along the beach, you're likely to see shells like these and might want to pick them up. And you might logically assume that they belong to animals that live in the lagoon. They don't. So these shells are 120,000 years old, and they've eroded out of the Feltre formations that you can see here. And so what you'll see on a little walk like this is a kind of a trip through time. So we've taken a trip through the landscape, moved across space. You can very easily move through time here at the lagoon as well by looking at the geological formations in these recent calcretes. These formations are 120,000 years old, and they are, where are they? There they are. They were deposited at a time when the level of the lagoon was the same as it is today. In other words, during the last interglacial, 120,000 years ago. None of these species occur in the lagoon at the moment. The water was a little bit warmer then. Here's pencil bait, for example, which doesn't occur here anymore. So conditions were a little bit different. So worth remembering that, you know, the environment changes not only through space, but also through time. What happened after that is that we got these fat deposits called the Langebaan Formation on top of the Feltre Formation, and these are terrestrial deposits of fossilized dunes. Effectively, they contain terrestrial snail shells, terrestrial gastropods. They are a testimony to the time when the great glaciers in the north sucked up all the water, and the lagoon dried up, and this area was covered by dune fields. The coastline moved out 30 kilometers offshore, and these terrestrial deposits were deposited. And then as the interglacial returned again, the lagoon flooded about 7,000 years ago, and we returned to quite similar conditions. So I'm quickly going to end with the one still unanswered question here, which is, where do the species come from in the first place? So most of the talk has been based essentially on the concept that you have a pool of species, the environment selects certain species from that pool based on their traits, and in that way the community is assembled. But where do those species come from in the first place? Well, they can obviously come from dispersal. We saw that just now. But many of these species are endemic to this region. Where did they come from? Well, obviously from speciation. So how does that work? As a quick example, I'm going to look at the oil-secreting orchids that grow here next to the lagoon. They're pollinated by oil-collecting bees that soak the oil up with modified front legs and feed it to their larvae. And those bees have these non-overlapping distribution ranges. And the one that occurs here in the lowlands, indicated with these blue stars, is Rediviva perengay, this top one. And it pollinates most of the oil-secreting orchids down here. But as you can see, there are other bees up in the mountain regions. So if you look at a phylogeny of the oil-secreting orchids, you see that that lowland bee with the blue stars pops up all over the phylogeny. So pollination by that bee has evolved many, many times. And you find in a phylogeny like this that sister species almost always have different pollinators. So the kind of scenario we're imagining is that an orchid would maybe disperse down from the mountains to the lowlands, adapt to the lowland bee, and as a result become a new species, which is now reproductively isolated from the one in the mountains. And if you transplant them, as we did here, between highlands and mountains, you find that the lowland orchid does very well at home, but does terribly up in the mountains. It simply isn't pollinated. The bees in the mountains just ignore it, even though they look so similar. And the opposite occurs if you transplant a highland orchid into the lowlands. It gets ignored. So this obviously has a lot to do with the scent of these flowers. Another famous pollinator of these lowlands is this extremely long proboscis fly that occurs around here. It has a proboscis longer than any known pollinator relative to its body size. And this fly, Magusterinchus longirostris, is endemic to the lowlands around here. And as you can see, plants on the lowlands have evolved super long tubes to match the proboscis length of that fly. And so if you compare proboscis length with tube length, you see the good match, and you see how long they are here in the lowlands. But in the highlands, the tubes and the tongues are very short, and this is a different species of fly. In fact, it's a different family. It's a tabanid. And so again, a similar scenario, dispersal onto the lowlands, adaptation to the local pollination climate, if you like, and then speciation as a result. This process has happened again and again, and has produced these wonderful guilds like this one of convergently evolved flowers that are in different genera sometimes, but have similar flowers with the same tube length. And these guilds of plants are, really one of the jewels of this area. They are amazing evolutionary products, which we need to conserve. But unfortunately, other people are looking for other jewels in the sand here, and these are proposed and approved mining operations on the west coast. You can see the red ones are already approved. Many of them are sand mines. They're looking for zircons and for titanium-containing heavy minerals. So you can see that as biologists, we're going to have our hands full in the next couple of years fighting these kinds of operations. Thank you.
Thanks, Anton. I'm sure some of you noticed that I just let Anton go into and through the question session because it was a fascinating talk. But the advantage of this being a hybrid conference is that you can ask him questions on the app if you're online. If your personal advantage is that we're about to go into a tea break, so you can catch him live and quiz him over a cup of tea and a pastry or the like. So we're going to have a tea break for half an hour now. So if you can all be back at 10:15, and Martina has a few announcements to make first.
That's going to happen a couple of times, sorry, but I won't take too much of your tea break. Just Madeleine said that the abstract booklet has been loaded on the app. Sorry, it wasn't there this morning, but it's available for anyone to view. If you haven't loaded talks for symposium following after tea break and in the afternoon, please do so. There's 1 or 2 talks missing. And I think I can do the others in lunch.
Yeah, I just also wanted to explain how the venues work. If you're all wondering where venue 1 and venue 2 are, you're in them. So this side is going to be venue 1, and this side is venue 2, and this is going to close. So if you've been wandering around the building trying to work out where the venues are, that's the setup. Cool.
Thank you. Enjoy your tea break. See you at 10:15. Okay. Hi, everyone. Welcome back after tea break, and welcome to the symposium, so Plant Life on Atypical Substrates. Definitely a very relevant theme to where we are on the globe. I'd like to introduce Sarah Palazzo, did I get that right? And Robert Boyd as your symposium organizers. And they'll be taking you through the next 2 hours with a couple of exciting individual talks. There'll be some time for facilitated discussion. And yeah, thank you, Sarah and Rob. We look forward to your session. Thank you.
Thank you.
Thank you, Martina. Thanks all for coming. It's very exciting to have so many people with us today. And we really hope you have a nice symposium. And we might be moving within Mediterranean type ecosystems. We're now thinking more about the soil and the substrate we are stepping on. So it might be a bit of a change in some regards. But we're going to start with our first speaker, Prof. Nishi Rayakaruna. He comes from California Tech University, correct me if I'm wrong, Polytechnic. And he's done an extensive work on serpentines, and he's going to be giving us an introduction of an overview on a typical substrate as a whole, because he's not only been thinking about serpentines, but also about how atypical substrates affect the evolution of plants. And he's worked intensively in California as well, and in other parts of the world. So thanks, Nishi, for being with us.
Perfect. Well, thank you, Sarah, for that kind introduction. Good morning, everyone. Welcome to the session on atypical soils. Karen and Anton, thank you for that great introduction to the region and to the ecology of the region. Really enjoyed it. This is my very first time in this part of the world. So I'm really excited I was really excited to get that overview of how cool this area is. Very cool, exactly. Today, and first of all, before I get started, I also want to thank the conference organizers and also the session organizers, Sarah Palazzo and Bob Boyd, for putting together a great lineup of talks addressing basic and applied research on plant life and lichen life and cross-kingdom interactions on atypical soils in the Mediterranean systems, but also more globally. Soils, as you know, is not only a medium for plant growth, but they're also really important in terms of shaping the diversity, ecology, and evolution of plants and their close associates, which includes things like beneficial and harmful microbes, pollinators, and herbivores. So plants that are growing on rock outcrops, island-like patchy rock outcrops with very harsh, chemically or physically harsh soils, like those found on serpentine outcrops or heavy metal-rich mine tailings or calcium-rich substrates like gypsum that you're going to hear a lot about today, have provided model systems to test ecological and evolutionary theory and conservation and restoration practices for a long time. So today, I'm going to focus on kind of the key lessons that studies on harsh substrate has given us over the last 50 to 75 years, focusing particularly on system studies that have come out of serpentine ecosystems. So as you know, climate is obviously central to the distribution and the diversity of species, but within a climatic regime, it is the geology that contributes to habitat heterogeneity and species discontinuity, like you see here on this photo taken from the Jasper Ridge Biological Preserve at Stanford University, where Hal Mooney used to work. I heard about Hal earlier on in the talk. Karen's talk. So what you see here is a serpentine outcrop and a sandstone outcrop that's side by side, which is a very common feature across California and some parts of the Mediterranean world. And what you see here is a Lastenia, which is in the Asteraceae, that dominate the serpentine outcrop. And then there are grass species, both native and non-native, along with shrubs that dominate the sandstone outcrop. This boundary is sharply defined and is maintained year after year. And when you do reciprocal transplant studies, it's very clear that things like competition with grasses keep the serpentine native or serpentine endemic species from encroaching the sandstone soil. And then some of the grasses that are more abundant on the sandstone do poorly on the serpentine because they lack the substrate tolerance. While geologic substrates that are very distinct, like serpentine and sandstone, are important in terms of maintaining distribution patterns in parts of California and other parts of the world, even within a geologic substrate like serpentine, you can actually find species that separate out along the outcrop based on their substrate tolerances to various microtopographical regions of a particular rock outcrop. So what you see here is two closely related congeners, species, Leucaena gracilis and Leucaena californica, that basically have a very sharp boundary. They are closely related and cryptic. They have a very close boundary based on where the soil conditions change. The Leucaena gracilis is more dry soil adapted, even though it's within the serpentine outcrop, and californica is found on the lower reaches of the outcrop, the swale, where there's a lot of leaching that brings in high concentrations of ions and increases the osmotic potential at the bottom swale of the outcrop. So just like distinct substrates are important in terms of creating really unique distribution patterns, even within an outcrop, you can find heterogeneity that contributes to unique patterns of plant distributions. When you do common garden and experimental studies and reciprocal transplant studies, you find that these species are locally adapted to different regions within the serpentine outcrop. The one that was on the top of the upper reaches of the soil outcrop is more drought tolerant, and the ones that are found at the bottom reaches are most ion tolerant. And reciprocal transplant studies show that they're locally adapted, even in this very small area of about 50 meters, they're locally adapted to their respective regions of the serpentine outcrop. Breeding studies and molecular work show that there's very reduced gene flow, both through flowering time and through pollen stigma incompatibility between these closely related species that look identical, but they maintain these distinct differences because of their very specific tolerances to the heterogeneity you find on this one outcrop. So this is one example, but there are lots and lots of atypical soils around the world. And what are atypical soils? So atypical soils can have one or more soil chemical or physical stressors, which include things like nutrient imbalances, high levels of heavy metals, very high or low pH, and also soils that have very poor structure, sand, rock, that ends up becoming very hard to maintain soil water content, and also for the roots to grow, especially if you have very clay-rich content soils. So soils are not just characterized by one harsh stressor. Mostly when you look at soils like serpentine or gypsum or other types of harsh substrates, you actually do find many of chemical and physical stressors that characterize that soil environment. So types of atypical soils. Today you're going to hear about serpentine or ultramafic rock outcrop soils that are generally high in heavy metals, very low in essential nutrients, has a really weird calcium to magnesium ratio where calcium is very low, magnesium is very high, and that creates a lot of stress. And in addition to that, you're going to hear several talks today on gypsum, which is a calcium and sulfur-rich substrate that also has led to really unique floras around the world, including in Mediterranean parts of the world. There are lots and lots of other atypical soils, saline flights, volcanic granite, gabbro, and soils in this region that I heard about today from Anton's talk, have nutrient poor. So all of those things, all of those types of substrates can be characterized as atypical soils. One of the common themes or features across many of these atypical substrates is habitat bareness, right? You have a lot of open areas with very shallow, rocky, clayey, or exposed soils that leads to high rates of evaporation. So thinking about plant traits associated with these types of habitats, you generally find traits that are what we would think of as kind of resource conservative strategy traits, where you reduce evapotranspiration under low water content. How do plants respond to atypical soils? Most plants exclude soils that are harsh. Those are called the excluders. Then you have the ones that are broadly tolerant, kind of the broad purpose genotypes that can tolerate both harsh soils and normal soils. Those are the indifferent taxa, or the bodenwalks, the soil wanderers that can cross back and forth. Then you have the ones that are largely restricted, which are the endemic species. And you see this across all of these harsh substrates around the world. So one of the systems that people have been looking at for the last 75 years is serpentine. And in 2011, myself and Susan Harrison from UC Davis came up with a book addressing how serpentine outcrops have been used as a model system to kind of study the effects of sharp abiotic and biotic gradients, patchiness and isolation, and harsh environments on ecological and evolutionary processes. We had 19 chapters that included 32 authors addressing serpentine as a model system to ask questions about ecology and evolution. So I'm going to kind of touch on 5 key lessons that we have found from studying plants that grow on ultramafic or serpentine soils. And one of the things that I'm hoping would come out of this talk and this session is to see how what we know from studies of serpentine outcrops applies to other types of atypical soils, right? So this session is special in that we are bringing people who are studying different systems to ask questions that we have been studying on serpentine outcrops over the last 5 decades or so. So one of the things that we know from studies of serpentine soils that generally places where there are serpentine outcrops, there are very distinct plant communities, right? And these communities may have, these outcrops may have low alpha diversity, but beta diversity is very high, regionally speaking, and these communities are generally very high in terms of endemism and rarity. If you think about California in itself, you have about 1.5% of California's landscape is covered by serpentine outcrops. So a very small region, but almost 15% of California's endemic species are restricted to serpentine soils. That's about 255 taxa. So, and if you look at latitudinally across California, you find that most of the endemics are in the northern parts of California where there's higher levels of precipitation, right? So high biomass, high competition of serpentine outcrops. So you have high specialization in this more extreme environment. Areas that have extensive serpentine outcrops that have a lot of heterogeneity, that also creates microhabitats for plants to evolve into. And then areas that have long exposure of rock, right? So the longer time, the more time for evolution. But what's interesting about California's flora is that it's not just serpentine that has contributed to California's plant diversity. You actually have, if you look at California's endemic species, about half of California's endemic species are specialists of atypical soils. They're found on gabbro, they're found on soils that overlay vernal pools, saline flats, alkaline flats, and all sorts of other types of harsh, chemically or physically harsh substrates. What's even more interesting is that, or of a point of concern, is that half of the rare, threatened, and endemic species in California double as specialists of atypical soils, right? So any kind of conservation or restoration plan or climate change models actually have to pay attention to the plant-substrate relationship to come up with informed decisions about how to move forward in terms of climate change. So one of the cool things about a conference like this and having a session here is that we can think about all of these Mediterranean habitats that we are familiar with that have a lot of endemics, a lot of really cool plant communities. But it'll be interesting to see how many of these endemics that you find across the Mediterranean region actually double as endemics of atypical soils. And I think this would be a cool place to start a discussion and even think about a project where we actually bring people from different parts of the Mediterranean region and look at the impact soil conditions, especially harsh, chemically and physically harsh soil conditions, have contributed to that level of endemism, and how many of those are of special conservation concern. So the lesson 2 is that a lot of these harsh substrates that are side by side with more normal or typical substrates are really good model systems to study evolution under ecological selection. Right? There's strong selection across these boundaries. There's sandstone on the left here, sandstone on the right, and serpentine on the left. And when you have these very harsh boundaries, you actually have potential for evolution of serpentine-tolerant genotypes that happen repeatedly across many parts of the California and other parts of the Mediterranean region. So here's an example that has been quite extensively studied, Collinsia sparsiflora in the plant Tegenaceae. There's intraspecific variation. There's white petal plants on serpentine and pink petal plants on serpentine and white petal plants on sandstone. They're morphologically very similar. And what you find is that hybrids within these ecotypes are infertile or sterile compared to hybrids within each ecotype. And which is a very common feature. You find these kinds of closely related taxa intraspecific variation across these substrates, where there's either through pre-zygotic or post-zygotic mechanisms, they're reproductively isolated. Sometimes there are morphological differences between these ecotypes, sometimes there aren't. What's also interesting is that these ecotypes or morphotypes are also colonized by different mycorrhizae, right? So to understand the role microbes or beneficial microbes play in kind of maintaining these kind of distributions is another area of importance in terms of study. Lots and lots of model systems have come up. Arabidopsis arenosa is one of those models. Monkey flowers is another model. And a lot of these model studies show that there's a lot of interspecific variation across plant taxa that are exposed to serpentine. Our crops here is a place where you're looking at two lineages of Arabidopsis arenosa. You see that when the serpentine origin plants are planted on non-serpentine soils, in the absence of competition, they do fine, but on serpentine soils, they do great. And when you bring non-serpentine origin plants and you grow them on serpentine soils, they are unable to do so. And this kind of intraspecific variation is really important in terms of generating the type of species that we have found as edaphic specialists across the world. There's a lot of cool evolutionary genomic work that's undergoing. And with respect to serpentine adaptation, at least we know that it is a multi-trait polygenic kind of basis for serpentine tolerance. The genes that are most likely responsible across these three different wild plant model systems is calcium to magnesium ion homeostasis, which is not surprising because serpentine soils are characterized by this weird balance between calcium and magnesium. Heavy metal detoxification and drought tolerance are important features that characterize serpentine tolerance across multiple wild plant model systems. There's also a lot of work trying to look at the pre-zygotic and post-zygotic mechanisms that are responsible for keeping these species separate. There are lots of examples of flowering time differences, shift to self-compatibility like Antonovics and McNeely found way back in the 50s with heavy metal tolerant plants. Hybrid sterility, reduced hybrid fitness, and pollinator shifts are really important in terms of evolution onto serpentine substrates. The recent study shows that heavy metals that are in the soil could directly contribute to reproductive isolation, because what happens is that when the heavy metals appear in the nectar of the plants, of the flowers, that selects for different pollinator guilds, right? So pollinators that are tolerant of heavy metals will start pollinating plants that are on serpentine outcrops that generally have a higher level of heavy metals in the nectar and avoid the plants that are nearby growing on a non-heavy metal rich soil. So serpentine and other type of atypical soil plant systems are really good examples to understand the genetic architecture of tolerance to substrate. And these wild plant model systems have been explored over the last 5 to 10 years. And they're also important in terms of understanding the factors that drive habitat specialization. The idea that why do only some plants become endemic to an atypical soil and others don't? So the idea there is that populations that are showing greater cost to tolerance might be the ones that might actually evolve as endemics to atypical soils. So in the serpentine literature, we see that there's a trade-off of competitive ability between serpentine-tolerant plants and serpentine-intolerant plants, in that plants that are endemic to serpentine soils are less competitive on non-serpentine soils. So that might be a driving force for specialization. There's also studies that show that there's increased susceptibility to herbivory of serpentine-tolerant plants. Serpentine-endemic plants are most susceptible to heavy herbivory on non-serpentine soils. Whether these kinds of factors are also important in terms of specialization of plants on other atypical soils is worthy of exploration. There's also meta-analysis of phylogenies that have happened in California with respect to serpentine-tolerant genera with serpentine-endemic plants. And what you find is this is an analysis that happened in 2011, looking at about 38 genera in California that have serpentine-endemic species. When you look at that, compare this taxa to look at the kind of the directionality of evolution to serpentine endemism, you find that many serpentine endemics in California have evolved from non-tolerator congeners or tolerator congeners, right? So there's generally a directionality in terms of evolution. There have been some reversals in some phylogenies where endemics have given rise to tolerator species, sister taxa, and then tolerators have given rise to non-tolerator species. And note that the thickness of the arrow represents the kind of the frequency of the observed transition. There's only one case out of 38 genera comparisons that showed a reversal between an endemic taxon giving rise to a non-tolerator, showing that becoming an endemic to a serpentine soil might perhaps be an evolutionary dead end. There are cases through integration and polyploidization. Some of these endemic species have given rise to other species, but very few of such cases available. It'll be interesting to see whether gypsum and other types of substrate that people are studying show similar trends across atypical substrates, where becoming an endemic is almost like an evolutionary dead end. Another thing that is important that I touched on earlier is that Plants that are found on serpentine and other harsh soils have resource conservative strategies, low growth rates, low specific leaf area, and whether these kinds of traits have a common genetic basis, because a lot of these traits are found across other atypical soils, are worthy of investigation to see whether the same kinds of traits that we see across different atypical soils have a common genetic basis. One of the last points that I want to make is that serpentine plants, at least experimental studies show that serpentine plants may be less sensitive to climate change, at least with the work that's been done in California, showing that plants that have looking at serpentine plants that are across the latitudinal gradient from low to high rainfall, you find that serpentine tolerant plants or endemic plants do not change their specific leaf area even under high rainfall, where their congeners found off serpentine actually have greater surface area as with higher levels of rainfall. So this kind of points to the idea of co-limitation. Serpentine plants are growing in soils that are nutrient poor, so having higher rainfall may not necessarily give them the ability to respond with a higher surface area because they're constrained by nutrients more than by rainfall. Serpentine plants that are adapted to dry, hot conditions might also do well. Perhaps they might expand their ranges of serpentine outcrops under a more dry, hot climate. So those are the kinds of things that could be potentially done through experimental work to see what kind of effects climate change might have on these edaphically specialized taxa. And also to look at whether the patterns that we found with serpentine applies to other atypical substrates. So final lesson. Sorry, I said final earlier, but this is the final lesson. Atmospheric deposition is a big problem in Mediterranean ecosystems, right? So the slide that I showed earlier from Stanford shows the serpentine outcrop in its prime state. You have the Lasthenia dominating the serpentine outcrop in 1980. This is a photo I took in 2009, and the photos obviously clearly show that there's a reduction in the yellow flowers that on the serpentine outcrop, and this has happened across California and some other parts of the Mediterranean region. The part of the reason for this, at least in California, is there's high levels of nitrogen deposition to atmospheric deposition, wet deposition, and dry deposition of nitrogen that has basically increased the nitrogen levels on serpentine outcrops, which has then made the serpentine outcrop susceptible to the invasion of grasses, especially Eurasian grasses that have come in, that were on the sandstone to begin with, but they are now adapting or growing on the serpentine outcrop. And these grasses have basically not only kind of contributed to the reduction in native plant diversity, but they have also resulted in the near extinction of a butterfly species that is reliant on some of these plants that are growing on serpentine outcrops. So, whether nitrogen deposition is also a problem in other types of atypical soils is something that we need to be looking at. And also the suppression of grazing and fire management practices like use of phoscheck, which is essentially fertilizer being dumped in an environment like California where fire intensity and frequency has gone up, is changing the nitrogen levels on some of these outcrops, which has also led to some problems in terms of invasive plant invasions. Information gaps, we need more studies, especially in tropical parts of the world. There's a lot of what we know about serpentine comes from the Mediterranean, California, and the true Mediterranean basin. There's a lot of cool cross-kingdom interactions that you're going to hear about from Dr. Boyd's talk in the next little while, next talk. Also, another interesting thing is that while there's a lot of endemism, high levels of endemism in vascular plants, there's not a lot of endemism in lichens and bryophytes. And this is a global pattern across serpentine. And one of the interesting things to potentially look at is whether the fungus is the same, but the photobiont is actually changing across its gradients. And that has not been really looked at, which might be an interesting area to investigate as well. Understanding the genetic basis underlying adaptation to atypical soils across different systems, the stress tolerance syndrome across different systems, those are areas that we need to look at as well. And also the relative importance of phenotypic plasticity and local adaptation, the work that Sylvia is going to be talking about and her lab has been doing on gypsum, is an area that has to be addressed in other atypical soils as well. Finally, these kinds of sessions need to happen, right? We need to bring people who are studying different systems into the same room to talk about patterns that they know from their own system. So I appreciate the fact that we have actually been able to do this. And as a motivation, as a kind of a result of this session, we've decided that we'll have another session on plant life on atypical soils next year in France, where the Serpentine Ecology group meets for the 10th time over the last 22 years to discuss research on plants that are growing on serpentine outcrops across the world, including in the Mediterranean systems. With that, I'm going to close, and thank you so much for your attention.
Thank you, Nishi. I think we've not enough time for
Just switch these. I'm going to change batteries.
Oh, okay. The batteries apparently are dying. At any event, I don't think we have time for questions, so we can stay on time. So hopefully on the chat or the conference, okay, Nishi will be able to do that. So I'm going to jump into my talk as soon as I see it, but there it is. And anyway, I'm going to follow on Nishi and talk just a little bit about plant-herbivore interactions. I have an extensive 5-minute time slot, so I'll be relatively quick. So my work primarily has been on serpentine, so I'm excited that we have gypsum colleagues here today. And there are a lot of differences, as Nishi mentioned, between atypical substrates and more normal ones. I'm going to focus on 2 of them today. One of them that's commonly used to define the differences are chemical differences on serpentine, calcium-magnesium ratio. High levels of heavy metals are commonly cited as major chemical differences. Gypsum, calcium, sulfur, also relatively high pH are a couple of chemical differences there. On serpentine habitats, there's another phenomenon that is interesting that I want to mention, and that's something called hyperaccumulation. On the graph on the right, hyperaccumulator plants, when they are grown on a low element substrate, have adaptations that allow them to take up extremely high amounts of a nutrient or an element and concentrate them in their tissues. So it turns out that many of these hyperaccumulators are found on serpentine and often hyperaccumulate nickel. About 75% of the 500 plus species of hyperaccumulators on the planet hyperaccumulate nickel, and more are being found all the time. You see the Science Daily from 2014. A tropical nickel hyperaccumulator was discovered and made the news. We're still adding to the list of hyperaccumulators. So why do plants hyperaccumulate? A colleague and I postulated that since those elements in high concentrations are often toxic, it might be to defend plants against herbivores. So we postulated that these could be called elemental defenses. Plants are defended typically by organic-based defenses, and there's thousands and thousands of those. But elemental defenses might be special or different in a couple of ways. First, they are made up of elements, which means that chemically they cannot be degraded. And secondly, they're taken up from the soil rather than made from photosynthetic products. So as a consequence of that chemical characteristic of the soil, there's a chemical characteristic in these hyperaccumulators, and there can be adaptations of herbivores, just as there can be adapted herbivores that are able to resist organic chemicals. There are herbivores that are able to avoid the defensive effect of hyperaccumulated metals. My students and I were studying a nickel hyperaccumulator in California and kept finding this little green bug. It was a specialist on the nickel hyperaccumulator plant, and we sent it off to the expert, and the expert said, hey, it's a new species, and they kindly named it after me, so I have to mention it in conferences everywhere, so everybody will know Melanotrichus boydii. It turns out the bug has a very high concentration of nickel in its body as well. 780 micrograms per gram is a lot for any insect. And so here's a consequence of the chemical constitution of the soil that leads to a chemical constitution in the plant, and in this case leads to a specialist herbivore with a high chemical concentration in its body. But of course, besides chemistry, there's another difference, and that's the visual quality of these backgrounds. I've got a serpentine and a gypsum dune example to illustrate those visual qualities. Color and texture of the background can be important. That can actually influence the appearancy of a plant to an herbivore. Nishi was mentioning how these are often open habitats. And it turns out a recent paper was documenting the different strategies for camouflage. There are 6 of them here. Animals have all 6, but look, plants have 4 of those. The top one, background matching, disruptive coloration of those seeds, basically masquerade, looking like what you're not. That is the second to the last one. And then finally, decoration. The idea that maybe even plants have little soil particles that could be decorating them and helping them be camouflaged in their background. Well, there have been some papers on serpentine. This is a paper on Streptanthus species that have illustrated that there is a signal of background matching between the foliage of these plants and the serpentine background where those populations occur in these different species of serpentine striptanthus. That's an interesting suggestion that background matching can be an anti-herbivore property that can drive plant adaptation. There's a second paper, also from the west coast of California, on a species that's found on and off of serpentine, and populations in the different kinds of substrates have different color patterns to their seeds that can also, through background matching, make them more difficult for herbivores, granivores, seed eaters to find them. So there's some examples here from serpentine habitats. I would like to know from my gypsum colleagues who are here whether or not there are any examples in gypsum, because gypsum has some chemical qualities that are fairly unique. Relatively high concentration of sulfates, for example. Can that be an anti-herbivore property? There's a paper by Sarah and al., Dr. Palacios's colleagues, that suggests that that can be an anti-herbivore trait. And then also the idea in gypsum habitats, if there's a visual signal, that that might lead to some anti-herbivore adaptations as well. So hopefully we'll learn a little bit more about that as the conference goes on. And I think that I've hopefully caught up on time so that I can end and let our next talk proceed. Thank you. Are we okay? On time? Okay, I think our next talk is a virtual one. So we'll have to get our colleagues. So would you like to? Real quickly?
Very quickly, yeah. Oops, sorry. So let me introduce Eric von Wendtvel from the University of Vermont in the United States. Eric was about to join us, but unfortunately due to health issues he couldn't make it, and he had to change to an online presentation, which is a real shame. But I'm sure we will enjoy it a lot. So I don't know if it's, yeah.
There we go.
There we go.
Hello, I'm Eric von Wepper. It is my pleasure to join all of you virtually. I'm very sorry I'm joining you virtually. I had every hope of arriving in person, and a bout of COVID between me and my family made that impossible. So I'm joining you virtually to talk about adaptation of crop wild relatives to extreme soils. I have featured on my title slide a picture of Nikolai Vavilov, the amazing Soviet-era Russian geneticist who came up with the idea of regions of crop domestication, Vavilovian domestication centers, where wild relatives of crops were selected, domesticated to be the crops that we rely on today. Some of the crops featured here are wild chickpeas and peas, both of which come from the Fertile Crescent Mediterranean ecosystem. So Vavilov's centers of domestication, they have shifted a little bit since he first coined the term in the 20s based on ideas from Candolle and Darwin and others. But if we look at where many of our crops came from, several come from partially or fully Mediterranean ecosystems in the Americas, in Eurasia, even in the highlands of Africa, in dryland ecosystems where there are large seeded grasses and legumes that have been the effective basis of our grain-based diets. So I'm going to particularly focus on some of the legumes. Interestingly, people everywhere around the world who have taken to agriculture have selected some form of bean to be part of their diet, whether it's a peanut or a groundnut from coastal South America, the beans from the Andes and Mesoamerica, soybeans from East Asia, some 20 species from South Asia, including pigeon pea, lab, and others that have been domesticated. And then in the Fertile Crescent, chickpeas, lentils, and peas that I'm going to focus on. In all of these regions, human selection had a huge impact on seed size, seed germination, toxicity of these species. But one of the things that we poorly understand is the ecology of wild relatives. And this is a shame because this is an important genetic resource for agriculture. Crop wild relatives receive almost no protection. We know less about their diversity than we should. And even more than that, we know less about their ecology, particularly their ecology on extreme soils. Given this is something that I've wanted to address, and we've been doing this over the last decade in chickpea, which is the second most widely grown pulse or grain legume. So it's a low oil legume like beans or chickpeas. Before our work were very few collections of wild chickpea, and most of them were repetitive. They had basically relabeled the same genotypes in different germplasm collections. So we went out in 2013 before IS became the problem that it has been, and we're able to collect across the Kurdish regions of southeastern Turkey. So this is just north of the Iraq border and the Syrian border. Often working with shepherds, working closely with Turkish colleagues who navigated the permitting and Nagoya consequences of our work. Our collections went into the Turkish National Gene Bank and have been available from there under the standard material transfer protocols. One of the sites where we find wild chickpea happens to be a serpentinic outcropping near a place called Maiden in Turkey. Maiden in Turkish translates as mine. So it's also a large copper outcropping in Eliezik province. So since I only have 6 minutes, I'm going to quickly thank my colleagues. I am happy to take questions by email. I'm easily found, and I look forward to hopefully seeing all of you in person one day in the future. Thank you.
Okay, thank you. Yeah, I guess we can send our questions to Eric. Now is the turn to Stefan Sievert. He's going to be presenting his talk. Stefan is also suffering from a long expedition across Namibia and South Africa that he's been driving some of us around, and now he's having back pain for that after many long hours of driving. So thanks, Stefan.
Thanks. Thanks a lot, Sarah. Yeah, so I'm a geoecologist from South Africa, and like Sarah said, we've just been on a long trip from all the way from Cape Cross down to Vredendal, surveying gypsum ecosystems. And yeah, so there you go. So when Sarah first approached me and she said, let's go and survey the gypsum ecosystems of Southern Africa, I was quite surprised, like many botanists, I guess, because we don't really give a lot of attention to gypsum in South Africa. In fact, the mapping system of the soil scientists in South Africa don't even include gypsum as one of the characters that they use to describe soils. So we did quite well, and we did find a lot of gypsum in these areas. But I'll be referring to it briefly to the expedition as I go through the talk. But about 3 years ago, we did a systematic review just to try and understand what's been done on gypsum in Africa. And because of the 5 minutes, I'm just going to focus on what we know in Southern Africa. Right, so as you can see on this map of Africa, the areas in gray are countries that have reported gypsum floras, or at least mentioned the occurrence of plants occurring on gypsum in Africa. So 17 of the 54 countries in Africa have already acknowledged that they do have gypsum plants. And then 23 papers of the 52 also reported that there are gypsum outcrops in their countries, and that's all the green dots that you see on the map. So a lot of the countries that do have gypsum have never ever even mentioned the occurrence of gypsum species occurring on these areas. So in the whole of Africa, most of the work has been done on the floras in the Horn of Africa, where there are massive gypsum outcrops. They are visible on the soil surface. But a lot of the work that has been done in these areas are focused on the description of new species occurring on gypsum, and very little work has been done looking at the ecology. But in Southern Africa, a lot of work has also been done. They found 17 papers looking at gypsum, not always directly stating that it's a gypsum ecosystem that they are working on, but deducing from the information that they provided in the papers. And a lot of the work, half of the work, has been on lichen fields. And I don't know if any of you are aware, but we have massive lichen fields in Namibia, and they also extend into the northern parts of the west coast in South Africa. And we've been sampling them, and quite amazing to see these areas. So just a couple of gaps that I noticed in the literature, which people have pointed out that we need to do more to better understand these ecosystems. The first one, we don't even have a list of gypsophytes for Southern Africa. So there's no list that lists the plants that have been recorded from these areas. So that's something that we need to work on. There's hardly been any work looking at how these species that occur in these gypsum ecosystems facilitate each other to be able to survive in these harsh environments. So there's a large gap of knowledge in that specific area. Then, although a lot of work has been done on the biocrust, the biological soil crust of the west coast of Southern Africa, there's been no specific studies just looking at gypsum and trying to understand how these soil crusts differ and what their function is. And then adaptive traits to try and understand how the communities are formed and how certain species dominate. What are the traits that make them more competitive? Hardly been, well, there's been no work done on that. So this expedition that we've been on, one of the main functions main outcomes of this expedition was to go and find data to try and answer these questions. Then also in terms of seed dispersal and how these species germinate on gypsum, very little work has been done. Very little work has been done on the lichen fields. So there's a reason for that, and I'll get back to that later. But lichens play a very important role in these ecosystems, and they've been neglected quite terribly in Southern Africa. Very little work gets done on lichens in the region. And then, of course, very little oops. Oh, now I'm completely out of, there you go. Very little work has been done, of course, on the evolutionary biology of plants that occur in gypsum ecosystems, which needs further attention. And then also to try and understand the biochemical cycles, understanding what happens to all the sulfates in the soils, how it's taken up by plants, and all the nutrient cycling. It's work that's never been looked at, and was also part of the Gypworld expedition to find information on this to understand the system better. So what are the reasons for this neglect in terms of research on gypsum ecosystems? The first one is that there's never been any specific projects looking at gypsum ecosystems in Southern Africa, of which the Gypworld project was the first one to really focus on these ecosystems. We have very little or no lichenologists, taxonomists in lichenology that are able to identify lichens for us in Africa, in fact. So we always have to rely on international collaborators to assist us with that. There are no clear maps, so we don't even know where the gypsum outcrops in Southern Africa occur. So that's a basic requirement that we don't have. And there's also no clear understanding in the botanical fraternity of what gypsum is. So we found many localities where botanists would report gypsum, that it was not in fact gypsum, and in times they say they were working on concretes, and then they actually collected plants on gypsum. Okay, so thank you. That is my presentation, short and sweet.
Yeah, I think we have time for one question, for one vice question. Someone there.
So, okay.
I just have a question in regards to the lichen fields. So they are very fragile ecosystems. Apparently, if you walk on them, your footprints remain there for up to 7 years. So how would you go about sampling such a fragile system and then also just taking out the human impact on the ecological findings which you would find?
Okay, so the soil footprint is not completely true. It's more if you drive over the lichen fields where you have an impact on the lichen fields, because the top of the soil is quite soft, but it can withstand the weight of a human, so you don't really sink into them. Okay, so, but yeah, we had a whole protocol that we followed to ensure that it's minimal impact on the system.
Thank you, Stefan. And now it's my turn, I think. Feel a bit like... Okay, I also have a flash presentation. So we are having this series of very short talks, and then we get into the longer and more detailed ones. And so I'll try to, in 5 minutes, summarize some of the findings we've had in our group about the nutrition of plants on gypsum soils. What is gypsum? We've been seeing a bit about it. It's a hydrated salt. It's made of sulfate and calcium and some water. And where we find it, so all over the world, over 200 million hectares. And to have gypsum in the soil, we need to have arid or semi-arid conditions. And that is why we find lots of gypsum in many Mediterranean type ecosystems, because they are somehow dry. It is an understudied substrate. It has been little studied as compared to other atypical soils. And this is particularly true out of Spain, where most of the studies on gypsum ecosystems have been undertaken so far. So we know really little, as Stefan has shown us, from other parts of the world, as for example, South Africa. So why is it harsh for plants to grow on gypsum? First, as we've said, because they are dry. I mean, gypsum gets leached easily with rainfall. So to have a soil with enough gypsum to affect the plants, we need the conditions to be dry. So that means plants will have little water to grow. But then they have physical and chemical properties that are special to them. And I've just summarized some of the chemical properties here. This is a table that shows the average composition of soils, gypsum soils in Spain. And we can see some of these soils have really high gypsum content. So we've measured up to 100% gypsum in the soil. That means tons of calcium and tons of sulfate. And that interferes with the uptake of nutrients by plants. So, and then it also leaves very little room for anything else to be in the soil. So these soils are very nutrient poor. Oftentimes we find phosphate goes below the detection limit in our soils, and they are also limited by nitrogen. So plants have to cope with this nutrient imbalance. But despite this, they are quite diverse, and as Nishi was saying, it's not the alpha diversity sometimes, but the fact that they include lots of rare and atypical specialized species. So over 1,200 plants across the world, we are trying to get a checklist for gypsum plants have been identified as having a high affinity for gypsum. In some parts of the world, like in Mexico, in the Chihuahuan Desert, gypsum endemics account for 7.5% of the total endemics. So in some areas, they can be quite diverse. And some of the research I'm going to present is related to how plants cope with this excess of calcium and sulfate in the soil. So if we look at the elemental composition of gypsum plants, for example, from the Iberian Peninsula, we can see that they differ. So we see plants with different gypsum affinity, so widespread gypsum endemics, and these are the plants that we think are very specialized to gypsum, because whenever there is gypsum within a region, they are there, and they only grow on gypsum. And we separate them from the narrow endemics, which are plants that also only occur on gypsum, but they have very narrow distribution. So it's easier, it's harder to tell if they are a specialist to these kind of soils. And then we have the tolerance. So the fact about the composition, the elemental composition, we really see that these widespread gypsum endemics have a difference in their accumulation of calcium, sulfate, and magnesium, for example, in Spain. So we wanted to know Why is this? What is determining the elemental composition of plants in gypsum ecosystems or in gypsum floras? And I'm going to present you here the results for the Iberian gypsum flora, but results are very similar for the Chihuahuan Desert gypsophilic flora. So basically, and this is not surprising, phylogeny is very important. So it's determining up to one third of the variability in the composition, in the elemental composition of these plants. But then, gypsum affinity, so how a plant has adapted to gypsum or not. It's also very important. So in the case of sulfur, this is for the whole ionom, but in the case of sulfur, it determines up to 10% of the variability in sulfur composition. So it's quite important. And we also see when comparing across different floras, like the Chihuahuan Desert, here in orange, and Iberia, we see that across different floras that have evolved on different climates, because in Iberia we have Mediterranean, but in the Chihuahuan Desert it's a monsoon climate. So rain occurs at different times, and lineages are very different. We always find this convergence in the accumulation of sulfur, calcium, and magnesium, specifically in white endemics. So why is this? Is there an ecological role behind this? We thought maybe it's because of the soil. So they are growing on soil that is rich in sulfate. Maybe that is why they are accumulating it. Well, we grew endemics and tolerance out of gypsum and on gypsum, and we saw that the endemics keep accumulating far more sulfur when they grow, even when they grow on a sulfur-poor soil. So they really have an affinity for these kind of elements. And we thought that maybe this is related to plant-herbivore interactions, following on works like Bob and colleagues. And we saw that actually, because gypsum occurs in open areas and they are subjected to herbivores, large herbivores sometimes, when we have grazing, and we have a grazing gradient, with increased grazing, the amount of gypsophiles in the community increases. So grazing is favoring gypsum endemics, and it is also favoring sulfur accumulation in the leaves of plants. So we think this can have a potential herbivore deterrent role that we have to explore further. So just to conclude, we find that gypsum endemics from different floras and climates, they tend to accumulate sulfur, calcium, and magnesium, sometimes over 2 orders of magnitude more than their relatives. This accumulation is not just the result of an effect of the soil, and in the case of sulfur, it could be related to an anti-herbivore process. And that's it. Bye, Tanki. Thank you.
Yolanda's up next.
Yeah. So I don't think there's time for questions, but you will find me over there. Our next speaker is Yolanda Puello, and she comes from my home institution as well, the Pyrenean Institute of Ecology.
Thank you, Sarah. Good morning to everybody. I think this is the last fast presentation. Later, we will have more time on each presentation. My talk is focused on grazing effects on gypsum ecosystems. I will present some results obtained in northeast Spain in a gypsum outgroup of this area. So, ecosystems and specifically rangelands, that are the topic of the talk, can be seen as plant soil systems with many components, all of them interrelated and affecting each other. For example, vegetation productivity, which is a very relevant component of rangelands, is highly affected in arid areas by the water availability for this productivity. And not only, also is affected by nutrients. These components of the soil are interrelated among them, but also are affected by the vegetation itself. The presence and composition of the plant community determines the quantity and the quality of the litter, and also it improves the structure of the soil to allow more infiltration and water retention. Grazing affects directly when these communities are grazed that affect directly to many of these components. For example, by direct plant consumption, by soil trampling, usually decrease water infiltration, and changing the nutrient pool of the soil by changing the amount and quality of the litter or directly through the excrements of the animals. And we could consider that indirectly the whole system will be affected by grazing animals. As Sarah nicely introduced, the gypsum rangelands are located on arid lands and share with the other dry lands the harsh climatic conditions. High aridity and extreme temperatures are always present on these gypsum areas. But the gypsum substrate add more stress to plant life with both physical constraints like the physical or biological surface soil crust or the water retention in the soil, which is usually very low. And also the chemical constraints mainly related to this unbalanced element composition of the soil. Well, grazing effects on rangeland has been studied for many years. It's well known, the effects on arid rangelands and semi-arid. But studies on gypsum areas are much scarce. But given that these ecosystems, these rangelands, present both highly specialized flora and physicochemical gypsum, special physicochemical properties, we can expect that grazing effects on these systems could not be similar or in all the aspects than the rest of drylands. So the objective of our study was to identify relevant plant-soil relationships and the effect of grazing on them in gypsum rangeland. We performed the study in the Middle Ebro Valley, which is a large gypsum outcrop, one of the largest in Europe, and quite pure, with usually more than 50% of gypsum content in the soil. We selected grazing gradients with low, medium, and high grazing intensities in 6 independent farms on gypsum areas. We sampled vegetation and soil variables in 1 square meter plots on these gradients. And specifically, we studied plant cover and biomass, soil hydrophysical properties like water infiltration capacity in the soil, bulk density, water retention in the soil, soil fertility, and soil microbial biomass. And we employed the structural equation modeling in order to detect the relevant interrelationship among these variables in the system. Well, I'm going to briefly present 2 independent studies. The first one, and given the relevance that water availability has for the productivity of the rangelands, was focused on the grazing effects on the hydrophysical properties of the soil. And what we saw very briefly is that the bulk density and the water infiltration, and more specifically some parameters determining the water infiltration, like the hydraulic conductivity of the intact soil, were the most affected variables by grazing. We also saw that gypsum content, the percentage of the content on each site, highly was affecting the same 2 variables. But the effect on these variables were the contrary than the grazing animals, and in general tended to increase the properties of the soil related to the water availability. When we put all together in this plant soil system the variables, we detected that the most relevant effects of grazing were affecting plant biomass and water infiltration in the soil. And these variables were affecting positively fertility and microbial biomass. In turn, hydrophysical soil properties were affecting productivity of the vegetation. But if we have a very quick look at the grazing gradient, we saw that the trends were not totally linear, but low and moderate grazing intensities still kept relevant properties for the plant soil system. So just to conclude, we saw that gypsum content is a relevant soil property on these systems, with cascading effects on soil and plant properties, and that moderate grazing can keep relevant properties in the plant soil systems, but increase the vulnerability because plant cover or biomass maintain the ecosystem functionality of the whole system. Thank you for your attention.
Thank you, Yolanda. I think, unfortunately, we don't have time for questions, but over the next break, I'm sure. Yeah. So now is the time for Silvia Matesanz. She comes from King Juan Carlos University in Madrid, and here's your microphone. Thank you. It's this one.
Thank you so much, everyone, for coming, and good morning, everyone. Today, I would like to show you the latest results on our latest experiment on rapid evolution on a gypsum street plant, Lepidium subulatum, from gypsum outcrops in the Iberian Peninsula. As we know, we've heard before this morning, gypsum habitats house very rich floras, and many of these species are endemic, and these habitats in the Iberian Peninsula are associated to climatic conditions that are semi-arid, and which are becoming more and more extreme over time, with the prediction of an increase, and a strong increase in temperature, and a strong reduction in precipitation. So there is different ways, different active responses that these plants, gypsum endemics, can show to respond to climate change. And today, I would like to focus on the ability of a gypsum endemic plant to show rapid evolution in response to the recent climate change that is still ongoing and that is predicted to become even more extreme over time. We have the notion, the intuitive idea, that adaptation takes a long time. Plants can respond really rapidly through adaptive plasticity, for instance, but for selection to act and lead to an evolutionary change, our intuitive idea is that it is a long-term process. But we also have evidence for both plants and animal species, different populations, that it is possible to have contemporary evolution at ecological scales. And with this experiment, we test precisely this, the ability of a gypsum endemic plant to rapidly evolve in response to the recent climate change that is already ongoing in the Iberian Peninsula. So there is different ways to test for rapid evolution. And we use a novel and very interesting approach, which is called the resurrection initiative. So let me walk you through how this works. Imagine that a botanist in the past, in an expedition of these gypsum habitats, like we just did, collected seeds. When he was in the field or she was in the field, and they collected seeds. And they did it in a way that they stored these propagules, these seeds, and noted down the coordinates of where these collections were done. So if we do that, or if they did that, and then we know these coordinates, we can come back to the very same place where these seeds were collected and collect these seeds again. Because over time, all these microevolutionary mechanisms may have occurred in this population. Of course, there might have been migration, mutation, drift, but what we are looking for is the action of selection. So if we get the chance to have these ancestors and these descendant genotypes from the very same place, we can then grow them together in common conditions, because comparing these ancestral and descendant genotypes would give evidence of rapid evolutionary change, contemporary evolution. And this is what precisely we did. And of course, if we think of this experiment to actually get robust inferences on rapid evolution, it is a long-term process. We need to have the collections properly stored. We need to have a measure of genetic variation. We need to control maternal effects, storage effects. So it's a long-term process, of course. But we did this with a very good, very fitting model, which is Lepidium subulatum, and we tested whether a population of this gypsum endemic from the Iberian Mediterranean gypsum outcrops has evolved to this ongoing climate change. And it's a really good fitting model because we know that migration via seeds is very limited for this species. So we know that microevolutionary processes that occur in situ are going to be key. We also know that both in experimental and natural conditions, this plant is able to reproduce as early as in the second season. So it has short generation times, which is key for this type of approach. And we also know that it, although it can live up to 25 years, one individual, we know that most individuals live for 6 to 8 years. So all these factors make it a very fitting model to test for rapid evolution. So we implemented this approach in a population in the central Iberian gypsum outcrops. And I'm sure you're wondering how we got a hold of the ancestral genotypes. And in our case, it was pure serendipity because the director of one of the germplasm banks in Madrid was an expert on the Brassicaceae, which is exactly what Lepidium subulatum was. So he would go just in the field and collect seeds from many different species from this family. And he did it in a way that these genotypes could be resurrected because he noted down the coordinates and he collected seeds from more than one plant, which is obviously what we need. So we were lucky enough to get seeds from 1964. And then we came back to the very same place in 2018, which is 55 years later, which is a pretty good time for this species. So we got our seeds, and then we performed a refresher generation. And this had a twofold goal. The first one was to minimize these storage effects of the seeds that are old, of course, but also the maternal effects that come associated with collection in different years, right? Then this refresher generation was also used to create genetic lines, different families, maternal families. We did that also separating them so that they were pollinated within collection year so that they were not mixed. And finally, we did some microsatellite analysis to ensure that the ancestral and descendant genotypes had similar levels of neutral genetic variation and low differentiation between them, to be able to disentangle the effects of these neutral microevolutionary processes compared to selection, to adaptive processes. So once we had our samples from the 1964 and 2018 of these ancestral and descendant genotypes, we did a common garden. We compared them. We wanted to see whether there were differences in their phenotype, in their reproductive biomass, in their fitness, right? So we created this common garden, and we had 2 different treatments related to the increased aridity that has ongoing in the last years. And we had 2 different treatments that reflect this variation in natural conditions related to aridity, which were well water optimum conditions, and then drought. And this approach allows to test not only the evolution of the mean phenotype, but also the plasticity of these traits, which is really interesting. And of course, we predict that if there has been rapid evolution in this population, the descendant genotypes would show higher fitness, particularly in dry conditions. So what did we find? In these graphs, what you see in the x-axis is the 2 different treatments. In the y-axis, you see a phenotypic trace, in this case, reproductive output as a proxy of fitness. And then the blue line, or the green line, is the ancestral genotypes, and the orange line shows the descendant genotypes. And we were really surprised with these results, because you can see how fitness, reproductive fitness, is almost identical in drought conditions, right? However, we found significant differences in the reproductive biomass of these ancestral and descendant genotypes in common conditions. And we were really surprised about this. But not only this, but also we found associated changes to the phenotypes. What we found is that particularly under well water conditions, the descendant genotypes, the 2018 genotypes, had lower lead dry matter content and also higher relative growth rates, which is consistent. We can interpret that as the evolution of more acquisitive phenotypes in these optimum conditions. So if we combine these results together from the phenotype and reproductive fitness. What we propose here, what we suggest, and these are preliminary results, so we would love to hear about your opinion because we're just thinking about this. What we propose is that what has occurred here is the evolution of higher opportunism. So these guys are not increasing fitness in stressful conditions. They are maximizing fitness when conditions are good. And this make sense in the context of the strong habitat environmental heterogeneity that the Iberian gypsum habitats and Mediterranean ecosystems in general experience. For instance, this is just in the study side, this population that we study, patterns of precipitation annually in the last 3 decades. And you can see how precipitation varies from year to year very, very much. And maybe this heterogeneity and this unpredictability is becoming more and more extreme. So the same genotype can experience a very, very dry year, and the next year can be much, much more optimum. And also, we have evidence for not only this gypsophile, but other species, that the differences in fitness between one bad year and one very, very good year can be from 2 to 10 times higher, which means that it may make sense to optimize fitness when conditions are very good, because for a perennial species, lifetime fitness may be much, much higher overall. And what we suggest, finally, is that success and adaptation to climate change in these gypsophiles may not involve evolving higher fitness when conditions are low, because maybe that's not possible when conditions are really bad, but to optimize and exploit very well what we call the favorable optimum conditions. And this is actually has been described in many other species, and it actually has a name, which is called the master of some strategy, optimizing the exploitation of really good conditions and maintaining fitness when conditions are really low. So to finalize, what we found is that contrary to our expectations, because this was, as I said, surprising, ancestral and descendant genotypes of a gypsum endemic collected almost 55 years apart had similar reproductive output in drought conditions. However, descendant genotypes showed significantly higher fitness in favorable conditions. This higher reproductive output was accompanied by evolved changes in several functional traits we're still analyzing. So we suggest that rapid evolution of an opportunistic strategy, maintaining fitness in stressful conditions while maximizing fitness in favorable conditions, may be what is going on here. And this strategy may have evolved in response to increased heterogeneity and climatic conditions, rather than to a change in the mean temperature or precipitation, which is something that we need to take into account. So our results suggest that rapid evolution in response to climate change may be possible in gypsum endemics, which can be crucial in species with limited dispersal. Thank you so much for your attention.
Thanks, Silvia. You really stick to the time. We have time for questions.
Thank you. That was a really nice talk.
Thank you.
So for local adaptation, you would expect those reaction norms to cross, right?
Right.
So do you think you may get them to cross if you used other conditions other than water? And what do you think that might be as a treatment?
Right. So I'm going to put a pin on your question, because in 2 more talks, we're actually talking about this, one of my students. And it's a research that we've been doing precisely to answer that, the interaction of drought, because we're focusing on drought, but we know there's more, warming and also other biotic processes like competition. So we are looking at what the norms of reaction do when you have combinations of stresses that are actually important in the field. So yeah, but yeah, I don't want to spoil it, sorry. Yeah.
You did a specific experiment with certain species, but do you have any idea about sister species as far as diversity in that system? Because in other systems like the dry forests, you get one species that sort of has a steady reproductive output, and then you have a sister species that really takes advantage of those good years.
Right. That makes complete sense to me. We don't know yet. So that would be something really good to see. The thing is, with this approach, it's really hard to get the ancestral genotypes. There is actually a project, baseline project, which is like a call to all researchers to collect seeds, not at the level of these banks that are Millennium Banks, but just for research. And I know that the collections are increasing and becoming larger, but it's something that, I think when I did this experiment, we were thinking if only I had collected all these seeds years ago. So, but for sure, we could do it just like with current genotypes to compare these different fitness strategies. And what we see in the field is that all of them tend to have much higher fitness when conditions are good, but they do it to a different extent. So for sure, there's something going on there. We'll look into it, yeah. Thank you. That's it. Oh, there's someone else out there. I don't know if we got it, if you want.
Yeah, I also really enjoyed that talk, thanks.
Thank you.
Yeah, I mean, an obvious evolutionary process that'll be running over those 50 years would obviously be adaptation to the gypsum itself, right? So how do you eliminate the possibility that they just haven't got better at increase their their fitness, absolute fitness on gypsum through time, through the chemical adaptation to the gypsum itself?
Right, so you mean that it's not evolution to climate, what has happened, but to the gypsum?
Yeah, absolutely. I mean, that pattern you showed is very common in all arid plants, right?
Right.
Reproduction in good years is what matters.
Yeah.
Reproduction in drought years never happens.
Yeah. I mean, we don't know. Of course, we don't know. But we did a phylogeography study on these species, and we were able to date the origin of the species, and it was like 3 million years ago or so. So I would say that if this species had 3 million years to evolve in gypsum, 55 years should not make more, evolution to gypsum more than to changes in climate. I mean, I feel like the selection pressure now is not the gypsum itself for this species, but the changes in climate. That would be my guess. Thank you. Oh. Yeah, sorry.
I'd like to ask about the possibility of nitrogen being a factor in this change through perhaps nitrogen deposition.
Right. We haven't considered that. For sure, it could be something that, yeah, and we could, we have more seeds because these in 1964 were so precious to get that we multiply them. So we could definitely try to make a plasticity experiment with different levels of nitrogen in the soil. That would be interesting. Yeah, we grew them in gypsum soils, which I didn't mention, but of course it was the same for all with the levels of nitrogen that they have. Thank you for your suggestion, for sure. Ay, Dios mío, hay muchas más. Yeah.
Hi.
Yeah.
Yeah.
I think it seems like we are out of time. Maybe we can talk later. Yeah, I would love to. Thank you so much, everyone. Thank you.
Yeah, and actually because we're going to be seeing results from the same group in the next talk. So, Mario Blanco.
There's no time for my presentation. Okay, good morning. My name is Mario Blanco Sanchez, and today I'm going to present some results from my PhD, in which we assess the role of adaptive differentiation and plasticity patterns in the dominant gypsophile Lepidium subulatum. In the Mediterranean region, drought is one of the main selective pressures for plant species, or in other words, is a critical factor that plants have to cope with. Furthermore, due to climate change, the climatic conditions will become harsher in the future, especially in areas with arid and semi-arid climatic conditions, such as gypsum habitats. These habitats harbor a very rich community of plants that can only grow in gypsum soils, named gypsophiles, and these species usually lack effective dispersal mechanisms. So one question of our time in evolutionary ecology within the Mediterranean region is how gypsophile populations will respond to climate change. And answering this question is particularly important to predict the survival and persistence of gypsophiles in a climate change context. During the evolutionary history of gypsophiles, widespread gypsophiles have been subjected not only to the evolution in a restricted substrate, but also in different climatic conditions, being an important selective pressure for the differentiation of populations. Therefore, assessing past selection events, whether plant populations have adapted to past climatic conditions, and the phenotypic plasticity patterns of these populations is key to predict and to identify populations adapted to their local climate or highly plastic populations that may have the ability to cope with the new environmental conditions imposed by climate change, but also to identify vulnerable populations to these climatic conditions. Despite this importance, it is unknown whether past climatic conditions have shaped adaptive population differentiation and the phenotypic plasticity pattern in Mediterranean gypsophiles. A particularly useful and interesting approach to assess the role of neutral and adaptive processes in population differentiation is FST-QST comparisons. FST is a metric that evaluates population differentiation in neutral molecular markers, and it quantifies the role of neutral evolutionary processes such as mutation, migration, or genetic drift in the differentiation of populations. In contrast, QST is a metric that evaluates population differentiation in quantitative traits, and it quantifies the role of neutral processes, but also natural selection in population differentiation. And comparing both metrics, we can assess the role of these neutral and adaptive evolutionary processes in population differentiation. When QST is higher than FST, different phenotypes have been favored in different populations during the evolutionary history of this species, suggesting spatially divergent selection across populations. In contrast, when QST is lower than FST, similar phenotypes have been favored in different populations, suggesting homogenizing selection. And finally, when QST is not statistically different from FST, population differentiation has been caused by neutral evolutionary processes. Using this approach, we assess the role of past selection and the role of climatic differences in genetic population differentiation and the plasticity patterns of Lepidium sublatum, and specifically, we answered the following questions. Is there evidence for adaptive population differentiation in key functional traits? If so, is population differentiation related to climatic differences between populations? And finally, are patterns of phenotypic plasticity influenced by past selection? Our study species was Lepidium sublatum, that is one of the most dominant and widespread gypsophiles in the Iberian gypsum habitats. It is a perennial shrub endemic of the Iberian Peninsula and North Africa, and it is mainly at crossing with partial self-compatibility. We sampled 11 different populations from 4 different regions, covering the entire geographic and climatic range of the species, and at each population we sampled seeds and fresh leaves from 20 different maternal plants. And from this data, these maternal plants were genotyped using 10 nuclear microsatellite markers, and from this data, we calculated population differentiation in neutral markers, DFST. Collected seeds were shown in 6-liter pots filled with gypsum soils, and after 2 years growing in common well water conditions, plants were assigned to each of 2 watering treatments that reflected contrasting watering levels, water availability levels in natural conditions. These watering treatments were successfully implemented using outdoor rainless clustering structures, and this was key not only to assess the phenotypic plasticity patterns of these populations, but also to assess the role of past selection in population differentiation under contrasting conditions, which is key to robustly interpret the results of FST-QST comparisons, since the expression of genetic variation is environmentally dependent. During the experiment, we measured a wide set of functional traits related to phenology, morphology, physiology, and fitness of individuals. And as you can see, we found that populations were remarkably different in their size and biomass, but also in other key functional traits that I'm going to show you. To assess if these phenotypic differences were associated to neutral or adaptive processes, we first calculated population differentiation in neutral markers, DFST, and we found a moderate population differentiation. Then, using Bayesian inference, we calculated the QST values for each trait represented in the y-axis in each watering condition. And finally, we compared FST and QST values using Kruskal-Wallis non-parametric test. And we found a significant genetic population differentiation in morphological, physiological, phenological, and fitness traits in both treatments. But more interestingly, in all cases, we found that QST was higher than FST, which reflect past divergent selection across populations. Or in other words, different phenotypes were favored in different populations due to natural selection. But what caused these differences among populations? Are these differences related to the local climate of the populations? And the answer of these questions is yes. Population differentiation was associated with the local climate of populations. And as you can see, hot and dry populations showed more biomass than conal humid populations, but again, we found significant genetic differentiation associated with the local climate of these populations in other key functional traits. These plots represent the main results of our trait climate associations, and in the x-axis, there is represented the an integrative climatic metric, the soil water content that was calculated using long-term temperature and precipitation data of each population size. In the y-axis, it is represented the phenotypic values of each population represented by each dot and in both watering conditions. And what we found, and as you can see here, is that populations from hotter and drier sites, that is, populations with lower values of soil water content, showed lower leaf nitrogen content and specific leaf area and higher water use efficiency and reproductive biomass, suggesting that past selection has favored a conservative strategy in this stressful environment to cope with these stressful conditions. Thus, we propose that genetic population differentiation was caused due to past divergent selection associated with the climate of these populations. And finally, the phenotypic plasticity analysis showed that populations responded plastically for most of the traits evaluated, but surprisingly, all populations showed similar plasticity patterns for most of the traits evaluated, as you can see in the parallel norms of reactions of all populations in all these traits. These results suggest that past selection may have shaped plasticity patterns, favoring genotypes with broad environmental tolerance in all populations, probably due to the high environmental heterogeneity of gypsum habitats in all of these populations. Just to sum up, divergent selection have shaped the patterns of population differentiation, being different phenotypes favored in different populations. Differences among populations were associated with differences in their climatic conditions, and climate was an important selective force during the evolutionary history of populations. Natural selection has favored a conservative strategy in populations with stressful conditions. It is with dry and hot environments, and phenotypic plasticity patterns were strikingly similar between populations. Selection may have favored genotypes with broad environmental tolerance due to the high spatiotemporal variation of gypsum ecosystems. Thank you so much.
Thank you, Mario. I think we have time for one quick question. Thank you so much. Okay, now we are going to move to Marina because, yeah, we had a last time cancellation. So, Marina Ramos, please. Thank you. Ah, okay. Yeah, sorry, this is the one talk that has been cancelled, so we have to move to the next one.
Good morning. My name is Marina Ramos, and I am a PhD student at Universidad Rey Juan Carlos in Spain. I am going to present some preliminary results from my PhD. Ah, okay, okay. Sorry. Vale. Okay. Drought is the main selective pressure in the Mediterranean region, and recent climatic models predict a significant increase in the intensity and duration of droughts, which are expected to be greater. Oh, sorry. Drought is the main selective pressure in the Mediterranean region, and recent climatic models predict a significant in the intensity and duration of droughts, which are expected to be greater in areas with semi-arid conditions. Therefore, assessing the mechanisms involved in adaptive responses to drought is key to predict the survival of plant populations. And this is particularly important in species with specific edaphic requirements and low dispersal ability, such as gypsophiles, plants restricted to gypsum soils. To assess the response of population to environmental change, experimental approaches with common gardens are usually done. In these common gardens, we can separate the genetic and environmental components of the phenotypic expressions of the population, allowing us to test adaptive differentiation among populations and to quantify phenotypic plasticity patterns. These common gardens can be used to predict the response of populations to future environmental conditions, for example, by identifying populations that are pre-adaptive to drought or that show high plasticity. Most studies with common gardens are usually performed with a single factor, for example, water availability. These experiments provide very important information on how population will respond to a single factor and are widely used in conservation to identify potentially vulnerable or resilient populations. However, we know that in natural conditions, gypsophiles experience multiple abiotic and biotic factors, for example, warming and competition. The combination of species may modify adaptive population differentiation and plasticity to drought. Therefore, the ability of gypsophile populations to cope with climate change must be determined by evaluating the phenotypic responses of population to complex and multivariate environments. When combined, multiple stresses may induce additive, synergistic, conflicting, or even antagonistic responses. In this graph, you can see that plants that experience drought usually reduce specific leaf area. However, in presence of competitor, plants may show higher specific leaf area. But what happens when plants experience drought and competition simultaneously? It is not clear what the response will be in the presence of both stresses. More importantly, the responses of population to drought may be affected differently by the presence of the other stresses. In other words, populations can respond differently to one or more simultaneous stresses. For example, as we can see in this graph, in response to drought, all populations reduce their fitness in the same way. But imagine this. In the presence of drought and the other abiotic factor, for example, warming, population differences disappear. And we will conclude very different things. And this is very important to make inference for the conservation of populations. With this example, I want to highlight that plastic response of population differentiation may be affected by a combination of stresses. And therefore, we need to assess them in multivariate setting, not by extrapolating responses to individual stresses. The main goal of our work is to assess the effect of warming and competition on adaptive population differentiation and plasticity to drought. We expect that phenotypic responses to drought will be affected by warming and competition, and that this effect will vary across populations, with key implications for prediction on climate change persistence. Our study species was Elentemon squamatum, because it is one of the most dominant shrubs of the Iberian gypsum. Elentemon squamatum has shown strong declines in reproductive fitness in dry and hot conditions, and lower survival in highly dense patches. This species flowers during summer, so it reproduces when environmental conditions are hard. We sampled seeds from maternal plant in 6 different populations, covering the geographical and climatic range of species. You can see in blue the selected population, and in yellow the distribution of Elentemon squamatum. We pulled seeds from each population, and we saw these seeds in pots with gypsum soils. After 2 years growing outdoors in common conditions, individuals were assigned to each of 8 experimental treatments, resulting from the factorial combination of 2 abiotic factors, water availability and temperature, and 1 biotic factor, intraspecific competition. Our watering treatment had 2 realistic and contrasted levels, well water and drought, and we implemented this treatment using a rain exclusion extractor. Temperature was manipulated using open-top chambers, which had been frequently used in warming experiments, or watering treatment simulated the average prediction of warming in climate change scenarios. Finally, in no competition pots, 1 individual plant was grown alone, and in pots with competition, 1 target plant was grown with a competitor of the same, on the same species. In each experimental plant, we measure a wide set of traits related to phenology, physiology, morphology, and fitness of individuals. But today, I am only going to talk about 1 relevant trait, reproductive biomass, that is the main fitness component. The results are presented using norms of reaction that show different treatment in the x-axis, reproductive biomass in the y-axis, and each line represents of population. We found a significant effect of drought, warming, and competition in the reproductive output of populations. All species cause a reduction in fitness, indicating that they are important selective pressure for our study populations. In addition, In addition, our results show a significant interaction between watering and warming, and also watering and competition, indicating that the presence of multiple other stresses have a combined effect on the phenotypic of individuals. Specifically, we found a synergistic effect in the phenotype, because individuals that experience drought and also warming decrease their reproductive biomass more intensely than those that only experience drought. Sorry. Also, we found significant differences between populations in their reproductive output. If you look at the norms of reaction, all populations, you can see that are very different. And these differences are not random. These differences are associated with the local climate of populations. As you can see, cold and humid populations in blue showed lower fitness in all combination of treatments, highlighting risks for this population in climate change context. And the most relevant results is that this population differentiation was lower when plants experience drought and warming together. As you can see, in the presence of drought and warming, the phenotypic differences between the driest and warmest population in red and all the other population were much lower. If we have done or experienced only manipulated water availability, our conclusion will be very different compared to an experiment with not only drought but also warming. And we see a very similar pattern when combined drought and competition. The driest and warmest population show a stronger reduction in reproductive biomass compared with other populations, even in the well water treatment, suggesting a potential trade-off in the ability of population to tolerate abiotic and abiotic stresses. To conclude, drought, but also warming and competition reduce reproductive fitness on the studied populations. These effects were not additive but synergistic. The presence of warming and competition strongly modified the phenotypic response to drought of populations. Cool and humid population consistently show lower reproductive output in all scenarios, which highlight the vulnerability of this population in future climatic conditions. Population differentiation in response to drought was strongly reduced in the presence of warming and competition. Our results highlight the importance of assessing the phenotypic responsive of population in realistic multivariate environments to infer their future persistence. Thank you so much.
Thanks, Marina. Unfortunately, we don't have time for questions, and we're going to move to our last talk by Sergio Muriel. Now we're going to move into lichens world.
Good afternoon. My name is Sergio Muriel, PhD student from Universidad Rey Juan Carlos in Madrid. My thesis project is focused on lichen communities from gypsum soils. Biological soil grass have a high presence in arid environmentals, including gypsum. They play important roles in driving ecosystem functions and providing ecosystem services. Lichens, which are part of these communities, are dominant organisms in gypsum soils and may reach a total cover of between 60 and 80%. Lichen participates in water retention, protection against erosion, germination of plant species, nutrient cycle, and as bioindicator of global change. With all this in mind, we pretend to understand how lichen communities from gypsum soils respond to global change, and more concretely, to answer two questions. The first one, which are the factors affecting lichen communities from gypsum soils along a climatic gradient, and which are the mechanisms permitting these communities to survive in gypsum ecosystems. In order to solve this question, we have studied lichen communities along a climatic gradient in the Iberian Peninsula. This climatic gradient is characterized by differences in temperature and precipitation. Climatic gradients has been considered as a value tool to understand global change. We have studied lichen communities from gypsum soils in 35 localities. As you can see here in the map of the Iberian Peninsula, here in Europe, and here are our localities. These photos represented several gypsum ecosystems that we sample. They are grouped in the north, central parts, and the southern parts of the Iberian Peninsula. We established plots of 30 per 30 meters in which we sample 10 subplots. Each subplot were sampled employing a square of 50 per 50 centimeters, and we estimated the total cover, of all lichen species present in the square. Each square was distributed 6 randomly along the plot, and 4 in each corner, as you can see in the image. Well, it doesn't matter. I can continue. Well, to study diversity, we calculate 3 facets of diversity. The first one, the taxonomic diversity, in which we calculate 3 different metrics: richness, inverse of Simpson, and Shannon. Concerning to the functional diversity, we analyze 2 different types of traits. Categorical traits, as for example growth form, if a lichen is foliose or squamulose. The type of photobiont is if it is or cyanobacteria or green algae. Or continuous traits, water holding capacity, or specific thallus mass, which may be related to the response of lichen communities to environmental factors. We calculated community weighted mean and raw index. And finally, we calculated phylogenetic diversity using raw index. We sequenced 5 gene regions of all species present in our study, and then we constructed a phylogenetic tree. As you can see here, it's small, but it's okay. We also employ climatic and local scale variables in order to assess the response of all diversities to these environmental factors. We obtain climatic variables from CHELSA. After we performed a Spearman test, we selected those variables that were not correlated among them. In this case, bio number 2, bio number 5, 12, 14, and we also included the aridity index. We performed some linear models in order to evaluate the relation between the diversities and the environmental factors. Unfortunately, we don't have enough time to include endothic factors, most specifically gypsum content. In total, 101 lichen species were present in our plots. The most abundant species were Squamarina lentigera, Diplosquistes diacapsis, and Sora decipiens. As you can see in our graphics, our community is dominated by crustose and squamulose lichens. Lichens with green algae as photobiont, and more specifically, lichens with secondary metabolites. You can see in the box plot that concerning two continuous traits, there are differences among species also. These are the results from our models. Orange squares represent negative relationship, while blues represent positive. You can see the three facets of diversity were significant related to the environmental factors in a similar way. Facets are negatively influenced by the mean diurnal range, that it was VO2, when the diversity is missing, when the diurnal increases. At the other hand, maximum temperature of the warmest month and precipitation produce an increasement of diversities. The taxonomical diversity is negatively related to aridity index. This produces resulting in an increasement of diversity when the aridity increases. And also we can see that climatic variables are more important than local variables. Here are the results from functional diversity and the categorical traits. We can see some interesting relationship between the traits and the environmental factors. The first one is that temperature variables are more important than the precipitation ones. However, it is interesting to note that they are differently affected to the traits. Maximum temperature of the warmest month and precipitation variables, an increasement of these variables, will result in an increase of the diversity of growth forms, being the community dominated by squamulous lichens, green algae photobiont lichens, and lichens with secondary metabolites. At the other hand, this suggests into a competition limiting similarity process, as in the general that I almost forgot to say it. At the other hand, an increasement of the mean diurnal range will result in a decrease of lichen diversity growth form, a dominance of foliose and crustose lichens and cyanolichens, suggesting an habitat filtering process. These are the results from the continuous traits that also you can show us interesting things. Not laughing, please. An increasement on the mean diurnal range will result in a decrease of all traits. At the other hand, we saw that maximum temperature of the warmest month and the precipitation will increase the diversity of all traits. However, we see our results show an opposite response to temperature and precipitation of traits concerning to the water use with this one with a photosynthetic performance. Maximum temperature of the warmest month will promote lichens with a thicker thalli, with high rates of water holding capacity and specific thallus mass, and a less photosynthetic performance, which results in a conservative strategy. Meanwhile, precipitation will promote lichens with a thinner thalli and less water retention, while and more photosynthetic performance, which results in a non-conservative strategy. Finally, our conclusions show us that the three diversity facets are related to environmental factors in a similar way. However, temperature and precipitation affect differently. Mean diurnal range will promote a habitat filtering process, while maximum temperature of the warmest month and precipitation will promote competition or a limiting similarity processes. In general, we can see that climatic variables affected or are more important than local variables, and we have to say that we are going to include it as soon as possible, Daffic and gypsum variables. And finally, I have to remark the importance of the role of the functional trends in the response to the global change. And we have to say thank you to everyone. Thank you for this month, that it was marvelous. Thank you to Prof. Stefan, Prof. Nisi, and other people that were included in our expedition. I had to say thank you, South Africa. It was a pleasure. And also thank you to the people that appear here, my supervisors, Isabel Martinez, Gregorio Aragon, and Maria is there, and to those guys that are marvelous. Yeah, of course.
I think when we look at the schedule, it says that lunch is starting now. Is that correct? At least the printed one says 12:15. Is it? I mean, okay, I just wanted to make sure. One of the rules of a conference is never stand between people and lunch. So I think we should conclude, and I would wish to thank everybody who presented. One more round of applause perhaps for them. They did a good job and they kept us almost on time. It was tough. And then we want to thank you all for attending. Y'all, we say that in the South of the US. Want to thank you all for attending, and we hope you have a great lunch, and we'll rejoin the conference at, 1:30, I think, when it's supposed to recommence. Oh, and then, yes.
Thank you, Robert and Sarah. Just one or two things before we break for lunch. Some important announcements. Sorry. And it'll go relatively quickly, but so any presenters for Tuesday or Wednesday, please upload your presentations. That's with the tech team or either through Madeleine from VetLink. Many of you would have noticed that there is Wi-Fi at the venue. It's the network is Athena, I think, and the password is Athena 2021. Please look for the banners or notices in the foyer if you wanted to connect to the Wi-Fi. There's a printed poster session this afternoon in tea break. It would be nice if many of you could engage with our printed poster presenters there. And then also the welcoming function this afternoon at 5:20. You didn't have to register or book for that, so that's open to all. Please join us for that. But the dinners for tonight had to be booked in advance. And tonight's dinner is at Cape Town Fish Market. I think it's from about 6:45 dinner. But the welcoming function is presented or basically in collaboration with Luby Rouge, and she'll be offering some local foraging material for some of us, or for all of us rather. Okay, so now we can break for lunch and we'll see you at Cape Town Fish Market, and to be back at 1:30, please. Thank you.